Okay, warm good evening to all of you. We will start the session and with yeah, we will invite the first speaker. Where is it? Professor Rana Pratap Singh, PHSS Foundation for Science and Society, Lucknow. Sir, please.
Please on your mic.
Yeah. Good morning, Honorable Chair, the organizers, especially Professor Dhankar, Professor Jose Puthur, and authorities of the society, and dear friends. I am here to share you some of our work and ideas related to designing ecosystems, below ground and above ground ecosystems, for better economic gain and ecological gain from agriculture. Friends, the restoration is a challenge now, but restoring the original ecosystem is not possible. We don't know what was there, what was below ground and what was above ground. So we have to design and we have to see what is going to ecologically successful. What kind of technologies, the gene technology, which has made it possible that we can bring character across the taxonomic boundary to plants, is a certainly very novel technology, but then plants has to work in ecosystem. So we have to see how it is behaving, how it is responding to the ecosystem, and how a new ecosystem is emerging by our own intervention and also by ecological succession. So on this some of our work I am going to share. So the emerging crisis we all know on this sustainable development goals are pollution, climate change, and loss of diversity, and so on. Then the new green economy is awaited, and we can see rather the whole economic developmental pathway is on stake, and the world is thinking that probably new green economy can be an answer in future. But a lot of work is required to be done to establish this new green economy. The poison in food bowl is there. We all know, we have been talking from yesterday about it, and a lot of contamination is there in our ecosystem, which is very rather dangerous, and our food system, water system, air system has been polluted. Our body is consuming a lot of poisonous things. So since nothing lives in isolation in the nature, whatever experiment we do in laboratory is required to be shifted to the field, and then we have to see the interactions there. Friends, the role of microbes are enormous. We have not talked about that, and the plant microbe interaction is required to be understood. In this connectivity, I have done some microbial modifications in soil and modifications in, rather than we have created some diversity in rather agricultural system above ground also, and I will show you our results, how it is responding in our case. So a lot of role are played by microbes. We have to understand, and we have to design their consortia in that way that maximum benefit can be taken, so that the chemical inputs in agriculture, which is backbone of the productivity in addition to seed quality, can be reduced. This idea, designer ecosystem, is floated long back, but then not many attentions were taken, and we recently wrote some review articles, and it was accepted by the scientific community that this idea can also be tested. Then the major challenge was to have some microbes which can be climate resilient, and plant assemblies, plant combinations and microbial combinations which are climate resilient. And here in India we have hot climate, drought and salinity and pesticides. All these are very pertinent problems in our agricultural fields. So we selected a very hot, very stressful climate, a plateau, to isolate some microbes, and this is from Ahmedabad, a plateau, dry plateau we isolated. And from metagenomics we found that there are, even in stony plateau, you can see in picture, there are 256 microbes of two groups, fungi and bacteria. Out of that we found 21 were culturable, and then they had these plant growth promoting activities. And these microbes had a good tolerance to salinity, drought, temperature, pesticide. And out of that we selected them and made some formulations and tested in laboratory conditions on wheat, which is a stable crop in India. And we found that the microbial combinations we are applying, which can be formulated in a consortia, and then with a carrier some entrapped bioinoculants can be prepared. And then these bioinoculants were tested in fields, and we found that the formulations are working well. These bound fertilizers are performing as good as the chemical fertilizer normally used in northern India, the urea and DAP. So then we made some multi-locational trials for these formulations in wheat fields, and then the similar results were found, and it worked well. Then another aspect we took, whether it is going to impact the quality of wheat. Quantity was found good, but then quality was to be tested, and for that we approached some exosomal inter-kingdom communication, how bacterial genes or bacterial inputs are influencing the plant expression of plant genes, and how plant genes are expressing the rather expression of consumer's gene, animal model. So here we tested and found that if chemical fertilizers are used and these microbial fertilizers are used, there are a lot of differential gene expression in plants, and we found that the about 1503 genes were different, and only 27887 genes expressed due to biofertilizers, which could not get assigned a known pathway. Means all genes which are expressed due to biofertilizers differentially from chemical inputs were not identified for their biological pathways. But then we made some studies and found that nitrogen metabolism genes and sucrose transporter genes, which are related to its yield and productivity, were expressed. And then some analysis, the exosomal isolation and its analysis conveyed that Yeah, it indicated that microbial biofertilizers promote the growth productivity yield of wheat. They can differentially regulate the genes and gene products related to nutritional value of the crop in comparison to synthetic chemical nutrients. These biostimulants can differentially regulate the genes and gene products of plants and animal consumers of the plants using an exosome related communication in comparison to synthetic chemical nutrients. Well, I am not discussing in detail, but we have indications and we are in process to confirmation that glutenine related genes are underexpressed during biological inputs in comparison to chemical inputs, and some cancer causing genes are also suppressed by biological inputs in comparison to chemical inputs. But since it is under confirmation, I am not emphasizing it. Then a lot of work on PGPR and PGPMs are getting done, but there are certain bottlenecks and why it is not getting adopted. Like phytoremediation also is not getting adopted in most of the countries at common level. In India and many countries are not able to adopt it as technology, probably because of certain field problems are there. Similarly here, there are certain field problems. Long-term field trials of multiple years on multiple locations are very rare. Studies on competition in inoculated PGPMs with native soil microbes are rare. Studies on their ecological successions and climate resilience in different agroclimatic ecosystems have rarely been studied. Effective mechanism for marketing management with user-friendly follow-up are lacking. Adequate promotional policies and regulatory provisions of governments are not available, and public is not sensitized on toxicity potential of synthetic fertilizers, pesticides, and other agrochemicals. Then we tried to create some above ground biodiversity also, in addition to creating below ground biodiversity, and certain endemic and these exogenously prepared microbial consortia were applied. And then again, why I say design, because we have to choose the plants. It is not just intercropping. We have to choose which kind of plant we want to grow in combination, and that require understanding of requirements of plant, emerging market requirement, emerging nutritional requirement, and also in addition to that, its suitability and resilience to the climate, agroclimatic conditions. So here we use Indian licorice, stevia, and chili. These three are commercial crops, and we found that the combinations, when we increase the combinations, biomass production and cost both increases with microbial inputs. Only microbial inputs are given in these studies, where means we kept the microbial inputs, same microbial inputs, and then we made variations in above ground diversity only. And here, this crop combinations label and PGPM's applications enhance dry biomass in a year, which indicate that more carbon sequestration and more biomass can be generated. And soil quality was improved very significantly. But it is not just straightforward that one plant, two plant, three plant, four plant. Means just by increasing the number of species, you will find all these benefits. It has to be basically we find in these cases. In this case, we find that all three are adding, but in other experiment, aromatic grasses, we found that only combination of two is beneficial. So we have to examine which kind of combinations, above ground combinations, and which kind of below ground combinations are beneficial. So here we then we rather calculated the carbon sequestration, and here also we found that if diversity will be more, certainly it is obvious that if below ground and above ground diversity will be more, there will be more carbon sequestration. The quality, all these crops have certain quality, so especially in case of this glycyrrhizine is the metabolite of this plant glycosine, and then its production was also quite better during the microbial combinations and multiple crop combinations. Then perennial aromatic grasses have high value and they are very suitable to marginal lands, very climate resilient. So their combinations were also tested. Vetiver, Symbopogon, and different species of Symbopogons were tested. Here also we found that combinations are performing better, especially combinations of 2, not 4, have given many good properties, positive outcome in first, second and third harvest. These are perennial crops, and obviously perennial crops reduce the expenses during the agricultural practices. Then species richness and PGPR application increase the soil microbial biomass carbon at different time intervals. It also increased the carbon sequestration in plant biomass, and then it increased the oil yield and agronomical and economical properties of the perennial aromatic grasses if the combinations below ground and above ground biodiversity were enhanced in a very designed manner. The different essential oils were tested, and it was found that the quality of essential oils, quantity was improved and quality was not changed because of these microbial inputs and above ground biodiversity enhancement. Then the same microbes, because solid waste management is also a problem, we tested, and this experiment is done in industry in Ethiopia, and we found that they are able to convert a very big amount of agro waste generated in avocado oil industry into fertilizers. So these microbes are of multiple use. We published the results, and this is my research group. So in conclusion, I can say that climate resilient beneficial soil microbes can be isolated from the rhizospheric niche and stressed agroclimatic zones, and their communities associated with suitable carriers can be developed as novel microbial products for sustainable crop cultivation. And another conclusion is the diverse compatible crop combinations consisting of emerging food and commercial crops for different agroclimatic conditions, nutritional and cultural diversity along with amended microbes can provide non-contaminated food security, high net return to farmers, and restore the degraded agroecology. Thank you. I think I am in time.
Yeah. Thank you very much, Dr. Singh. And we can take 2 to 3 questions. Oh, please.
Thank you, Doctor, for your nice presentation. I have a curiosity just. In the field trial with pepper, chili pepper, licorice, and stevia, did you notice an increase in secondary metabolites due to PGPR treatment?
Yeah, it is increased. Yeah.
Thank you.
Thank you.
I should have sat over here. Can I ask a question about the longevity of the plant growth promoting bacteria over time? Did you go back and test at regular intervals to see if the bacteria that you applied was still there after 1 year, 2 years?
Yeah, this is very relevant question, but we have not done it. Certainly the succession will be there, and in different kind of soil ecosystem, they will perform differently. That is required to be done. I agree. Yeah. We have not done it. Thank you.
Oh, wonderful presentation. And
thank you, sir.
Professor Singh, have you tried because there is a CSIR Institute in the Lucknow Central Institute for Aromatic and Aromatic Plants. They are trying to use the yield of the menthol. So I think it will be a good idea to increase the yield of menthol using certain potential microbes which you have isolated.
Yes, certainly, sir. This is a big institute, and there are certain philosophical issue. I can say I am working with a non-profit organization at present, and we have seen in certain areas that mentha they have introduced, and in those areas, especially Barabanki, water scarcity is there, and farmers say that when they started cultivating mentha for export and for whatever may be for increasing farmers' income, they were requiring 5 to 6 irrigation, and now they are requiring 15 to 16 irrigation. So we don't agree with the concept of exporting the agricultural product which are consuming high resources, and the resources in India are very rather less as per population, and this mentha cultivation is water consuming. So we have chosen the aromatic crops which do not require much water. And moreover, CSIR people don't give importance to universities and small groups.
No, that is not the case. We all come from the universities.
Thank you very much, Dr. Singh. And I would like to thanks for nice presentation. And our next speaker is Sri Bhas Roy from University of Hyderabad. And the title of the talk is Elevated CO2 driven population specific RNA directed DNA methylation pathway module, oxygen biosynthesis in Arabidopsis. Dr. Roy, please.
Thank you to organizers for giving me opportunity. Thank you.
Is in pen drive?
This one, right? I just know one, sir.
042?
It is. Like you can take.
Yeah. This one is for.
The pointer doesn't work because this is the LED screen, right? Because you can use the one.
Okay. Thank you. Very good morning to all of you. First of all, let me thanks organizers, especially Professor Jose, for inviting me to give a talk on this specified topic. My topic is, as you see, it is carbon dioxide driven population specific. I'll be talking about two populations which are growing at different environmental conditions of Himalayas. One at very low altitude and other one is very high altitude where extreme cold and extreme night and day temperatures is prevailing. The natural populations they have adopted there in contrast to low elevations where there is not that much temperature fluctuations or for that matter any light or environmental other factors. Then RNA directed DNA methylations, the small RNA that drive the DNA methylations. So this is a process where the methylation group is added to the DNA. So this process, what we found, is operating differently to these populations. And we'll see its consequences and how these two populations has evolved this pathway to cope up, maybe, we are not sure, to the existing environmental conditions. So that's the way my title defined. And let me go to the basic of this. There is an eraser. That means it will erase the memory from the DNA, and there is a writer who will again try to add that particular memory, and there is a reader who will only read that yes, it is there, it is not there. Accordingly, this combination of these three functions to drive the biological processes or epigenetic modifications in the plant and animal. And that's drive the adaptations what we are talking today may be responsible for some of the processes. So we all know that how elevated carbon dioxide has arisen from the before pre-industrial area to the till date what we see per se. And this graph is taken from the climate change web page. So I don't have to go in details of that. We are all aware of this rise in carbon dioxide in the environment. What we know is that we know all that India is one of the richest biodiversity hotspot in the world, and these are the specific groups, the species that are present that has been reported from India, including large number of angiosperms, which comes around 20,000 of these species. In spite of this biorichness and works on traditional botany, the model plant Arabidopsis thaliana might be knowing it has come down to now low from its peak utilizations that has helped us to understand biological processes enormously. And the first plant genome which was sequenced, unfortunately we didn't report this plant species from the Indian regions, and our group first reported that yes, in the Himalayan regions we do have these plant species, and we look into that how these plant species in the Himalayan regions may unfold some of the unknown facts of these processes. So these two populations, there are along the elevations, as we move from the low to high elevations, there are change in atmosphere, and the populations are also growing there. So we have a unique chance to explore direct effect of environment on these populations and how they have shaped their phenotypes or genotypes or epigenetics over the periods. And when we see that these populations, importantly, when we compared with the world populations, if we see world population has been exploited to its full extent, and you know now that if we speak about Arabidopsis funding agency, we are not going to give you any fund anymore. In spite of that, we tried our best to study these populations. What we found that Indian populations seems to be different from the world populations, both genetically as well as epigenetically. These data just summarized as fact that yes, Indian populations, we have compared 1,200 genomes of the world and our 50 genomes of our Indian populations, and it seems they are different from the world populations. And what is DNA methylations? Now, it is the adding of methyl group by the enzyme system in the plant to the DNA. And this is the process by which DNA methyl group is added to the DNA. And what does it cause? It can be added to different sequence context, CG, CHS, and CHG. H stands for other than C or G. Anyway, so this process I am not going to talk about that, how this small RNA adds DNA, methyl to the DNA. This I'll talk later on. And now what is the consequences? If this methyl group is added to the DNA or removed from the DNA, it can activate or suppress the gene and consequently its effect on the phenotype or whatever the gene does. So that is the effect of modifications of the DNA. Having said that, now let us come to our observation that these two populations were we hypothesized that these two populations, since their environmental condition is quite different, so they might respond differently towards elevated carbon dioxide. What we did, we grow these two populations in the normal conditions for 12 generations to standardize their whatever temporary epigenetics memory the methyl group may have erased or they are permanently fixed. So 12 generation is sufficient for Arabidopsis to erase its memory so that we come to a stable conditions. Yes, this population is stable, so we grew them in the normal conditions of normal environmental conditions, and then we subject them to the one group to the elevated carbon dioxide and one group in the normal environmental conditions. That is the phase facility, free air carbon dioxide in these facilities that is available in our CSIR NBRI lab where earlier I used to work. And then we morphologically and physiologically we standardize these populations. The physiological processes, namely photosynthesis, then transpirations, water use efficiency, both increases in both the populations under elevated carbon dioxides. But stomata, they do changes with respect to the elevated carbon dioxide conditions. Then we evaluated the methylome sequence of these two populations to see where the methylation changes do occur differentially in these two populations. And these are the data which shows in different contexts what is the DNA methylation level, in which context, in which sequence context these methylations are there, and what are the genomic context, whether it is in genic regions, whether it is in the promoter regions, whether it is in the intergenic regions, how they are differentially methylated between these two populations, as well as between the normal environment as well as elevated carbon dioxides. So what we observed is that this number of differentially methylated genes we identified between the populations as well as elevated carbon dioxides and the normal conditions. Then we identified differentially expressed gene for those two conditions. We call them population specific or growth specific. And then we identified what are the genes that are differentially methylated as well as differentially expressed. So those common genes are of our interest, because the gene expression as well as methylation is interrelated. So we identified some of those genes which are commonly differentially methylated as well as differentially expressed. Then we categorize those genes which are mostly involved in epigenetics pathway, namely chromatin modifications, then DNA methylation, and small RNA biogenesis, because these are the three processes of epigenetic process. And we identified those genes which are mostly involved in these processes. So subsequently, what we did, we validated that yes, what we have observed, that differentially methylation, do they really exist or not? What we do, we did them as one methylation inhibitor, that is azacitidine. If we grow the plant under in a media containing azacitidine, the methylation process is inhibited, and then you can understand that if there was methylation or not, and consequently its expression also changes. So by these two combinations, we can validate that yes, there is a methylation, and then there is a gene changes in the gene expression. We do say that yes, the changes has occurred. So this azacitidine treatment and subsequently expression analysis, and then you follow the bisulfite sequencing. The three process conclusively says that yes, your DNA is methylated. You have the treatment that you have given, it caused the DNA or it methylation, or it removed the methylations. There one gene, I'll talk about one gene. This is THB4, which is tryptophan synthase B gene. Now we call it I will use these two genes for, in recent term, it is P5P genes. So these two terms may be interchanged together. So this THB4 gene, which was found to be both differentially methylated under elevated carbon dioxide only in one of the populations. That is, in the low elevation population, this gene was found to be demethylated when it grown in elevated carbon dioxide. Again, we do validate. Yes, you have given elevated carbon dioxide, the gene has been demethylated. So we do probe that yes, this gene is demethylated when you grow in the elevated carbon dioxide, and when elevated carbon dioxide is not there, the gene is again remethylated. So this gene, we consider that this gene has something to do with this elevated carbon dioxide as well as its adaptations at the specific environmental condition. And the gene was found to be involved in this pathway in the last step of tryptophan biosynthesis. And tryptophan biosynthesis, tryptophan is the precursor for auxin biosynthesis. What we observed is that after validation with bisulfite sequencing that yes, the gene is methylated and demethylated, we then grow them under the demethylated plant. We again grow them in the normal conditions that it reappeared, methylation reappeared. So it is not stable. It is only demethylated under elevated carbon dioxides. This bisulfite sequencing as well as transcription analysis as well as gene expression analysis do suggest these methylations and demethylations. Oops. So how we know that this methylation is methylated or demethylated? So we carried out small RNA sequencing. That small RNA might be responsible for, and then we found that one of the particular small RNA was found to be targeted to that particular methylation demethylation site. We identified that this small RNA does complementary to that specific methylation site. More importantly, when the expression of this small RNA is higher, then demethylation rate is lower. So consequently, we do found that yes, this may be responsible for one of the methylation or demethylation in this pathway. This is a complicated pathway, but what I would like to draw your attention in is that the H3K9 methylation, that is histone modification. This histone modification, this methylated histone is a mark. It can be identified by the RNA polymerase, and that leads to the small RNA biosynthesis, and these small RNA do drive these methylation modifications, and we then validate it by using with or without these demethylated regions to change to see the expression and methylation, and we do found that yes, indeed, when this promoter is with the small RNA and without the small RNA region, then there is variations in the methylations, and when we do the immunoprecipitation with that particular histone modification, we do find that small RNA precipitate with the histone antibody, suggesting that this small RNA is responsible for demethylations of the gene, and then we quantified the auxin biosynthesis that is responsible for, and we expressed that the TSB overexpressed genes, there is much higher synthesis of auxin, which leads to these modifications in the plant's biomorphology, leading to early adaptation, early maturity, and we suggest that this model suggests that under elevated carbon dioxides, this histone modification mark is removed. And
Dr. Roy, please conclude.
Yep, that is my conclusion. Thank you. And this is my team. And thank you. Thank you for.
Thank you very much. And we can take one or two questions only.
Excellent presentation, Professor Srivastava. So, using these small RNA pathways, do you think we can make the climate smart plants for the, especially climate smart crop plants to sustain the productivity in the time of the elevated carbon dioxide conditions?
Yeah, that is a burning questions. Always we are asked whether the model plant system can be applied to the crop plants. To specifically answer these questions, we need to understand as much as we can from the well-known processes so that these processes can be exploited in the higher plant or agricultural crop plants. Yes, these processes, if we understand that, yes, why this model plant? Because it gives us resources to understand. Now, if this pathway do exist in the agricultural crop, which is more likely, then I believe this can be. You can just find out that this small RNA is there or not, and whether this gene is methylated or not under that conditions. If that is existed in that particular crop of interest, we can do manipulate that.
Thank you.
Dr. Jason, please.
Very elegant work. So CO2 clearly elevated CO2 clearly induces this methylation. Do you think other abiotic stresses would as well?
Yes. Yes, all abiotic stresses, there are lots of work.
It would be interesting to look at them in combination, elevated CO2 plus temperature exchange or something like that.
Yeah, temperature, that is also has been done differently, but because elevated carbon dioxides do rises the temperature as well. Mm-hmm. So combination of these we have not evaluated, but people have done in other for other traits, other specific conditions. There are lots of paper, not with respect to these particular DNA methylations and histone modification. This is the second report. This is not, we have not published, but our publication was second. First one was from a group of Stavinia in Italy, I believe. That group was first group that reported the methylation changes in response to elevated carbon dioxides, and subsequently we also reported this in one of our publications. And this pathway, what we are showing and what we have observed is unique, and it has implications in the combination of that abiotic stress tolerance.
Yeah, very elegant work. Nice.
Yep. Thank you.
Thank you very much, Dr. Roy. And our next speaker is Dr. Neeta Lakda from Chaudhry Charan Singh Haryana Agriculture University, Hisar, and the title of the talk is Role of OSRLK gene in enhancing salinity tolerance in pigeon pea, insight from omics approach. Dr. Neeta, please.
A very good morning to all of you. First, I would like to say thanks to the organizer for providing me this opportunity. So, the topic of my talk is role of OS-RLK in enhancing salinity tolerance in pigeon pea and insight from the omics approach. So,
please adjust the mic.
As we know, the world population is increasing day by day, and land for the agriculture is decreasing because of number of abiotic stresses such as salinity, drought, flooding, and out of these abiotic stresses, salinity is the one of the major abiotic stress which affect the crop, plant, and development. And other effects of the soil salinity is the loss of infrastructure and reduction in the water quality, and also cause the soil erosion, and further the wastage of energy required to desalinate it. And in India, almost 6.73 million hectare area is affected by the salinity stress. So we need to, scientists look to develop the salt tolerant genotypes which can be grow in cultivars which can be grow in the saline area to feed the increasing population. So in this study we have used the peas and pea plants, which is a most important legume crop of India, and India produces 66% of the world peas and pea production. As we know, this is a very much important legume crop. Legumes are very high in content of protein, carbohydrates, sugars, and various vitamins and various trace elements, calcium, iron, magnesium, manganese, and zinc. And it is also having high dietary fibers and low glycemic index, and it's a gluten-free and high in antioxidants and high in energy, and also anti-diabetic and anti-cancer property. So, but these legumes are very much sensitive to the salinity stress, and the plant respond to various ways to adapt to these salinity stress conditions. First, by accumulating the various osmolytes and various antioxidative enzymes also enhance. And one of the major process is affected by the salinity is the photosynthesis, and decrease in the chlorophyll content and carotenoid content observed during the salinity stress conditions. And also this salinity stress is also reduce the nitrogen supply from the soil to the plants, and which lead to the lower the protein content in the crop, and finally it leads to adversely affect the biomass, pod filling, and the yield, ultimately yield of the plants. So scientists all around the world are trying to develop the saline salt tolerant crops by using various methods, conventional as well as the genetic engineering. But conventional breeding approach is very lengthy, as we know, and labor intensive and costly, and depend upon the excess of the germplasm, which need the various genetic variability. So to resolve these barriers associated with the conventional breeding, the most effective way to develop cell tolerant plants by genetically engineering the desirable gene in the plant. So here for this study we have used the LEC-RLK genes, so lectin receptor like kinase. As the lectin receptor kinase family is a group of receptor like kinase that play several vital roles in the plant growth and development, pathogenic defense, symbiotic association, and different abiotic stress responses. And this figure shows the signaling pathway, how does this LEC-RLK receptor like kinase pass the signal to the final output. So this LEC-RLK structure consists of one of the extracellular lectin domain, a transmembrane region, and a cytoplasmic kinase region. LEC-RLK located at the plasma membrane, which can transduce the signal and trigger the plant defense and the stress response. Upon perceiving the extracellular signal, the LEC-RLK undergo the autophosphorylation, and that even lead to cascade of various phosphorylation of the intermediate protein phosphorylation. And these intermediate proteins can either continue the cascade by the downstream protein phosphorylation and various activation of the transcription factor and hormonal pathways also modulation, and ultimately leading to the activation of the stress responsive or the developmental signal, which leads to signal responsive genes. But few studies nowadays are available with this lectin receptor kinase role in the stress tolerance. Earlier it is reported for the biotic stress, but now the studies are focused for the abiotic stress also. And these are the various reports which are reported in case of Arabidopsis, rice, and Pisum sativum, and Nicotiana tabacum. And one of the recent study from our group by Mehla et al., which also shows that the OS LEC-RLK overexpression in peas and pea plant demonstrates show the superior performance in respect to the wild type from the vegetative up to the reproductive phase and compared to the wild type plants. And this study is mainly in continuation with that work, because in that work we have published various biochemical and the morphological and physiological aspect and yield parameter, but the study need to understand the molecular mechanism behind it. So this is the construct of the vector map and then agrovector transformation. This is just a pathway which we have followed by the protocol during this development of the transgenics. And further in this study we will discuss the protein and metabolite profiling using the LCMS and GCMS to understand the molecular mechanism which is still unknown or not fully understood. So proteomics, as we all know, the proteomics and metabolomics is a very powerful tool to understand the molecular mechanism. So protein are the functional product of a gene and involved in metabolism, cellular development, and but also have a role as a toxin or anti-nutrient or allergens also, which have a great impact on the human health. So it also proteins also provide stress tolerance to the plant. So proteomics provide and also it is also provide information about the unintended changes while transfer of the gene. It also provide information about that one. And another approach is metabolites, which are defined as the study of metabolomics is study of the whole metabolites present in the cell. So metabolites are defined as small molecules developed or modified during the cellular metabolism. And metabolomics provide the information about the nutritional factors of the plant, okay, how the nutritionally affected by the salinity stress any crop plant. So it is necessary to analyze the characteristics of the transgenic plant before commercialization and also assess the potential unexpected consequences of the transgene to the human health. So in the present study we are mainly focused on the proteomics and metabolomics approach to uncover the various key protein and metabolite involved in this Le Carrelke mediated tolerance. So this slide is focused on the showing the differentially expressed protein. We have compared the wild type plant or the non-transgenic plant with the transgenic one, and we found that the thousands of proteins are differentially accumulated in both the cases. So but maximum when we followed this gene ontology process, then we found that maximum number of proteins are involved in various biological process, this metabolic process, biosynthesis of secondary metabolites, amino acid biosynthesis, carbon metabolism, and most of the protein localized in the cytoplasm and the plastic. This is the list of the differentially accumulated proteins, and here we can clearly see the proteins related with the antioxidative pathway and photosynthesis related process is highly accumulated in case of the transgenic, and this one of the most important highly accumulated protein in case of the transgenic during the stress condition is fumaryl acetoacetate hydrolyase, which is having a great role in the salinity stress tolerance. It is reported earlier, and calcium binding protein also. And further we did the KEGG pathway analysis for all these differentially accumulated protein, and we found that here we are mainly focusing main process of the cell. So here the photosynthesis related protein, and we found that the number of protein related with the photosynthesis are highly accumulated in case of only in the transgenic with respect to the wild type crop. And these are the protein table. Here we can see various cytochrome, oxidase protein, glycerol, aldehyde, okay. And one more thing we have reported, mainly in case of the wild type plants, various proteins related with the glycolysis is upregulated in case of only wild type plant with respect to the transgenic, and pyruvate kinase, fructokinase. And this is the pictorial presentation of all the KEGG pathway of all the proteins reported by the oxidative phosphorylation also. And these are the, okay, one more thing is the important proteins are the ribosomal protein, which are needed for the protein synthesis, and we found that these are highly accumulated in case of the transgenic plants, which is needed for the proper activity of our cell for the protein synthesis. And these are the list of the ribosomal proteins. And another most important pathway is the proteasome pathway, and where we can see the 26X proteasome and pathway is related protein with the proteasome assembly is highly accumulating during the salinity stress condition in case of the transgenic. So it is a very crucial, proteasome assembly is a very crucial cellular machinery responsible for the degradation of the proteins which are damaged during the salinity stress condition in the plants. And further, we extend our study to the metabolite analysis. And in case of the metabolite, we found that the various osmolytes related metabolites are the proline, alanine, serine, and the glutamic acid, and various sugars, sucrose, glucose, trehalose, and galactose and mannose are highly accumulated in case of the transgenic. And further, the various fatty acid and different categories we have observed in the fatty acid related protein which are also found to be highly accumulating because it is the membrane is disturbed during the salinity stress condition. So plant try to adapt to by accumulating the more osmolytes and the metabolites and the proteins related to with these pathways. And for the organic acid, here we can see the organic acid is more accumulated in case of the wild type plants. So this oxalic acid, coumaric acid elevate the saline alkaline stress. So various actually from this study we just get to know different pathway used by the wild type or maybe the transgenes, different pathways followed by for the adaptation to the stress. So in case of this when we do the metabolic pathway analysis, we found that under the stress condition in case of wild type, major pathway affected is galactose metabolism, glycine and serine threonine metabolism. But here we can see in the transgenics, various different other pathways are also pathway related metabolites are highly accumulated is glycine, serine, threonine, which are common one, and but starch and sucrase metabolism, and another is glyoxylase and decarboxylase metabolism, and galactose metabolism, which is high, and another one is the sulfur metabolism, which is highly upregulation in case of the transgenic. So summary of this work is protein expression and metabolite analysis help us to revealing the working mechanism of OS-like-RLK in providing the stress tolerance, and transgenic plants display this differing expression of the protein related with the photosynthesis, carbon metabolism, osmoregulation and protein degradation, because during the stress condition plant has to maintain their stability, and more accumulation of ribosomal protein which is involved the stability of our protein synthesis machinery, and protein involved in ubiquitination also increased during the salinity in case of the transgenic plants, and other amino acids, proline, alanine, serine, glutamic acid and various sugars were accumulated in case of transgenic, more accumulated with respect to the wild type. And although some significant observed in the protein and metabolite accumulation in the transgenic as well as compared to the wild type, but these proteins which are highly accumulated is not related with any allergic or all the proteins are not toxic. Not any toxic or allergic protein has been found during this study. And so finally, okay, and this result may provide information for further eliminating the potential effect of like-RLK in another plant crop species, and it can be explored for the human health or further. And this is the conclusion of my study. So we, as I have already told, the various proteins which are related with the photosynthesis and oxidative phosphorylation is increased in case of the wild type plant, and in case of in case of the transgenic plant, and in case of non-transgenic plant, various succinate glycolysis pathway and various proteins related with succinate dehydrogenase, NADPH. So more of the energy is going to be in the glycolysis and TCA pathway in case of the wild type during the stress condition. But in case of the transgenic, most of the shift of the energy pathway to the making the increase the photosynthetic and oxidative phosphorylation. So these are my lab members. Thanks.
Thank you very much, Dr. Neeta. And we can take only one question because we have already behind.
Okay, thank you very much for your talk. A quick question. You used the rice kinase, not
yes.
yeah, which was lovely, and I saw that gave enhanced tolerance to salt. I wondered about the endogenous P kinase. If you'd investigated that and upregulated it and see, did it have a similar effect?
Yes, in this study earlier we have published from Dr. Upendra lab, Mehla et al. He has also checked the endogenous expression of that gene. While the stress condition, it is increasing.
Okay. And
yeah, we have checked
so
it.
You don't know specifically what the difference is in the kinases between the different species, because you looked in tobacco as well, and that doesn't confer stress tolerance to salt.
But we have checked in silico also. It is showing the homology. With the various like RLK present in this study by Dr. Kuteja lab, and they have already explored this type of in silico analysis where they found the very much similarity between these like RLKs during these various crop plants. So that's why after that they have transformed in the overexpression in the rice as well as in arabidopsis, and so many crops they have checked. And our focus is mainly, we have to go to the legume crops. Legume crop, we can check it. How it is behaving. So it is providing stress tolerance in case of the legume also.
Okay, thank you. Lovely work.
Thank you.
Yeah, it is multi, since these stress tolerance, abiotic stress tolerance is multi-character regulated process. So just one gene transfer is helping a lot. So is it transcription factor?
No, sir. It is a receptor-like kinase. It's a signaling related protein actually.
Okay.
So while the stress, whenever the signal came, then signal pathway is activated and then goes to the various activation of various transcription factors and proteins which is related with the adaptation.
Okay, Neeta, congratulations. Good work.
Thank you, sir. Thank you, sir.
Thank you, Dr. Neeta. Now, I would like to invite our next speaker, Professor Om Prakash Dhankar, University of Massachusetts, MST, USA. And the title of talk is A Novel Gamma-Glutamyl Cycle for Improving Toxic Metals Stress Tolerance and Food Safety via Maintaining GSH Homeostasis in Plant. Doctor, please.
Some technical glitch. Please copying my slide.
Your time is start.
Session 3. Can you check whether this one is yours?
No. This is one, yes.
Okay.
Right.
Okay.
Go ahead, full mode. I'll move from here.
You can do this one as well. It's okay.
Okay, I also noted my time now. It's starting now. Okay. So, hello everyone, good morning. And I'm going to talk about this gamma-glutamyl cycles that involved in the glutathione homeostasis and glutathione, how the glutathione homeostasis provide tolerance to multiple stresses, and how this glutathione pathway could be utilized towards phytoremediation as well as abiotic stress tolerance in crops. And also, I want to put some of those fun slides. Our friends here, not in this conference, not that one, but my friend here, Jason, he had a blast in Chandigarh, that he was enjoying this motorcycle ride. Also, both Jason and Baushan, 2 years back, they came to Delhi with me. They hired this auto. I think Baushan is driving now, so he has a part-time job there. So anyway, so that's why he's not here today. So just to show that for fun that they had good time here, and I'm sure all of the foreign participants must be having good time here, and before and after the conference. Thank you. So the point here is now, many of the previous speakers made the point that abiotic stresses and climate change and global food security is a big issue. So here, as we know that, I will just briefly go through that. We know that this by 2050 we will be crossing 9.7 billion. We are already crossed 8 billion people on the planet, and because of this, the increasing population pressure, we need to produce more food, almost doubling the food price. With the limited resources, because the land and the this is something with the land. I will not use this one. With the land, decreasing land, lack of water, lack of fertilizers, and all those are making it a formidable challenge for the scientists to make more climate resilient crops and produce more for the farmers to grow more and produce more food. and that's become also, now we are shifting to the biofuel as well, and that's where the biofuel will be basically from the crops, and crops need to be grown on the same land, so there will be a competition between the biofuel crops and food crops, and then that could lead to the food price increase as well. So there is a huge challenge that to address that in the future. And also the focus on toxic metals, and all these toxic metals, yesterday I showed you that in this one, from all different sources, recycled water, wastewater, pesticides, and all those, everything get into the soil and in the crops, and crops accumulate those, and then we eat those crops and affect our health. And recently, the US government, US Congress, they commissioned 2 studies, and they found out that alarming level of heavy metals, arsenic, lead, cadmium, mercury, and chromium in the baby food formula, which most of the companies, they are labeling it organic food, baby food, and they had the 1,000 times more level of toxic level of these metals than the normal permissible limit, and that's how could it be organic in a way. So it's kind of like this is a gimmick by the companies and playing with the health of the babies and the population. And so in US, there is the focus on the closer to zero action plan, which means reducing those toxic matters in the food chain, especially in the baby food, to the zero level. And me and Dr. Jason White were basically engaged by the US Department of Agriculture to do a brainstorming workshop and write a report and submit a report. What are the limitations, how this could be addressed, and we submit that report, and the report is basically used by USDA to leverage more funding for the heavy metal toxic work. So in this talk, I will be talking about gamma-glutamyl cycle, and in the gamma-glutamyl cycle, what is the glutathione? There are 2 steps in that, the synthesis and degradation, and how those will lead to the enhanced abiotic stress tolerance. So glutathione is, we heard about those, many talks as well. Glutathione is the most important antioxidant in the cells, and also people take glutathione supplements as a spark of life. Even glutathione is in your beauty creams for enhancing fair skin cream kind of those. So glutathione is everywhere in those, whether you eat or those plants need or everything, everywhere it is there. So how the glutathione homeostasis is maintained in the plants. So in that, this is the complicated cycle, but I can show that this is the synthesis part of the glutathione here, and this is the degradation part of the glutathione. So in the synthesis part, when these 2 enzymes here, that's the cycle yesterday I showed you. So here, this gamma-glutamyl cysteine synthetase and glutathione synthetase, this slide got messed up. So that these 2 enzymes make the glutathione. And the first step is the rate limiting step. Gamma-glutamyl cysteine is the rate limiting step. When we overexpress this, the level of those this glutathione was the gamma EC, and glutathione was, what is this? Was significantly increased. And then when the glutathione level increased, we check the plants under the arsenic and mercury stress, and these are the wild type plants. And here they, at 250 micromolar arsenate, they did not, they germinated, they died, whereas gamma ECS overexpression plants are moderately tolerant. This is jumping somewhere. Yeah, so, and then same thing with the mercury as well. And then this increase in the glutathione level lead to a beautiful strategy for phytoremediation of arsenic. There is no one arsenic hyperregulator plant been identified, Teres vittata. It's not working everywhere. It only grows in the warm tropical climate, and it's not working in the cold climate. So we need a genetics-based strategy or portable strategy that we can put in those genes in any native plant species. So we use this glutathione enhancement approach, and we overexpress with one of these here. This is the arsenate reductase from bacteria. It reduces arsenate to arsenite. And if it happens in the roots, then most of the arsenite bound to glutathione and goes to vacuoles and store in the roots. But we put a tissue-specific expression way that we put on the RC reduction only in the shoot, so that reduction of arsenate to arsenite happen in the shoot, and glutathione increase has also already been improved the glutathione level in the shoot. So it binds to glutathione and store the arsenic in the vacuole of the leaves, above ground tissues, so plants can hyperaccumulate arsenic in the above ground tissues, and then we can harvest and recycle the metal and clean the environment. So we did that one, and this work was done, beautiful, elegant work was done with two former postdocs, Sudesh and Dr. Kundan Kumar. He's sitting in the audience, he's here, and the work is being carried further now by Joe, the graduate student sitting there, and then new postdocs, and putting several genes together. Like now we are stacking 5 or 6 genes together in those. So then they translate that into field crops, like what was the question was Dr. R.P. Singh was asking to Dr. Sribas Roy, that the translation from the model system to the real crops. And that's like one of the examples I will show you here. So when we overexpress these 2 genes in Arabidopsis, the wild type plant, so gamma-ICS plants are moderate tolerant. RC plant was very sensitive because it converts less toxic to more toxic arsenite, and gamma-ICS and this RC and gamma-ICS double transgenic plants become super resistant to arsenic, and they accumulate 3 times more arsenic in the above ground tissues. So per plant basis, it was almost like a 51 times more, 17 times more biomass, 3 times more arsenic. So per plant basis, almost 51 times more. Then we translate into cranberry, a non-food high biomass food crop. And here I can show you that when we translate this one, again slides are missing. Yesterday happened again. Anyway, so this one is missing something. So what we did was we transferred that. I gave you a different slide. I think you guys copied a different one now because it was different one there. So, and then what happened is when we translate into cranberry plants become super resistant to arsenic, these double transgenic plants, and they accumulate almost 2-fold more arsenic. So whatever the results we had in Arabidopsis was translated very well in cranberry as well. So then how the glutathione degradation step works. So now we showed the glutathione synthesis pathway enhance the arsenic tolerance with the more glutathione and also lead to the an elegant strategy for phytoreduction of arsenic. How the glutathione degradation happens. So this work was started by Bibin, a graduate student, and now another graduate student, Gurpal, carried over, and now the third graduate student, Oves, is working on that currently. So in this pathway, glutathione is basically degraded by this gamma-glutamyl cyclotransferases, GGTs, and there are 3 enzymes in those Arabidopsis, and then it converts into oxoproline, which is a precursor of glutamate, and then this oxoproline or pyroglutamate is converted to glutamate, and glutamate is recycled and then used into the for resynthesis of those the glutathione, and then cycle goes complete. So what we did here, we overexpress this one of the GGCT gene, and it leads to significant tolerance to arsenite, significant tolerance to mercury, and also to ABA for abiotic stresses. And knockdown lines were sensitive to ABA, sensitive to salt, and sensitive to mannitol for abiotic stresses. So basically, not only the increase or the overexpression of this glutathione degradation pathway that lead to the maintaining the balance cycle of the glutathione, leads to the tolerance to multiple stresses, abiotic stresses, as well as to the metal stresses. And then here what happens is, in the pathway, when you overexpress this gene, this will lead to the production of more pyroglutamate or oxoproline, and that was the level of high level of oxoproline in the overexpressed lines compared to wild type and compared to knockdown lines. And in this case, the glutathione level was not much changed because plants utilize it, recycle it, and balance cycle goes on that one. And that's how, so we are saying that overexpression of GGCT create more efficient gamma-glutamyl cycles to maintain the glutathione homeostasis and increase the tolerance to multiple abiotic stresses. And then also, this also can lead to the improved nitrogen use efficiency because glutamate is the first amino acid to be synthesized from the nitrogen assimilation, glutamine and glutamate. Those are nitrogen storing amino acid. If our plants can internally recycle glutamate, it can cut down some percentage of usage of the nitrogen assimilation from the fertilizers. And here we showed that overexpression lines, like here, when you knock down these lines, basically what happens is, you can hear me, when you knock down this gene, the glutathione is not degraded, so the thioxoprotein level goes down and the glutamate level goes down as well. So that means this plant requires more nitrogen from the environment. And that's the case when we use the labeled nitrogen, labeled glutamate, we saw that here the wild type plants, the assimilation of the labeled nitrogen from the external source, here is the mutant plants, and here are the overexpression. Overexpression lines has the minimum exogenous utilization of the labeled nitrogen. That shows that they have the internal recycling of glutamate. And when you overexpress, you can see these plants require less nitrogen from the outside. They maintain the balance internally as well. So this plays a role in the improved nitrogen use efficiency as well. And then transferring this technology to the field crops. And again, application. My lab is working on the basic research with the applied focus. Everything we do, we translate in those different crops. So here, we use the gene from the camelina, from the oilseed crop, and we overexpress this gene, and then these lines, overexpressed lines, these are a high level of overexpression, and these are the use, we tag with the DS red fluorescent marker. You can identify the transgenic lines with the UV light, the transgenic seeds, and the wild type seed does not glow red. So it's very nice way of detecting the transgenic line without going through the antibiotic selection markers. So here, when we overexpress this camelina GGCT2 gene, and we grow this on different arsenic stress, here is the without any arsenic stress, wild type under 3 transgenic lines, and here here under 20 micromolar arsenite, wild type plants are severely stressed, transgenic plants are thriving on that. So they're highly tolerant. And then these lines also had, it's changed everything. So these transgenic lines compared to wild type has higher biomass, and also that was biomass, root biomass and shoot biomass. When we analyze the total arsenic in these lines, they are highly tolerant. They had 70% less arsenic in the shoot, and also almost 50% less arsenic in the root. So these plants, they accumulate significantly lower arsenic. So if you use those lines, these genes in rice and other crops, they will produce a safer level of safer level of rice with less arsenic accumulation for food safety. So these could be, now we are translating into rice to reduce the arsenic accumulation for food safety and for human consumption. So with that, I can just conclude here that glutathione homeostasis stays, overexpression lead to the enhanced accumulation of glutathione that lead to the enhanced level of tolerance to toxic metals and elegant phytoremediation strategy for arsenic. A breakdown of that increase the tolerance to metals as well as other abiotic stresses and significant decrease of the arsenic accumulation. Now we are stacking all those genes in different combinations, different food crops for the reducing the toxic metal accumulation as well as in non-food crops for enhancing the accumulation of those metals for phytoremediation purpose. And that's my group here, and the funding is highly acknowledged from the funding from the NIH, the grant, and then the ARPA-E, Department of Energy, and USDA. And this work was mainly basically Sudesh and Kundan, and then the Gurpal and Bibin and Oves. That was the work done from there. Thank you very much.
Thank you, Professor Om, for being before time. We can have some questions from the audience.
Yeah, I have a question. Thank you very much for your nice presentations. It was really exhaustive work. I have a question that when you applied that 40 micromolar arsenic in Arabidopsis, there was lots of root biomass increased a lot. How do you see that, and is it with respect to arsenic or anything else?
Let me clarify that. When you applied arsenite, the root biomass does not increase in the control plants. It inhibits. Its root biomass increase in the transgenic lines which are tolerant to arsenic, not in the control plants.
So because we overexpress this gene, they have higher glutathione level. Glutathione bind to metal and store the metals into the vacuole, so that those metals arsenic is not interfering with the normal metabolic process in the cytoplasm. And that's how the tolerance mechanism works for the glutathione. It's a chelation kind of strategy, binding the metal with the glutathione. But the biomass or root biomass or shoot biomass increase was in the transgenic line only, not in the wild type. That's how I want to make it clear.
Why is that? Why the root was so happy, root biomass?
Because plants are tolerant. Because the plants are tolerant to arsenic. But because arsenite binds to glutathione, and once it's bound, it's taken out from the cytoplasm, stored into the vacuole, and vacuole is a secured compartment that vacuole is basically a landfill for the plant, that any waste or toxins are dumped into that one for preventing the toxicity of those metals in the cytoplasm. That's the tolerance mechanism. That's a well-known fact.
Sorry, quick question. Is there any crossover with selenium? Because they shared periodicity. So I wondered if selenium deficiency in your arsenic overexpression plants.
Yeah, there is the competition between silica and selenium. Selenium is less on that one, but silica is one of that, because arsenite transporter and silica transporter that Kundan was showing yesterday and Dr. Tripathi was showing some as well, those are same. So in this case, there are well-proven studies that if you have more silica, there will be competition between arsenite and silica. There are all proven that, but in this case, we did not check because it's already been done in many places. Yeah, thank you.
Excellent presentation, Professor Dhankar. You have used a combination of genes. Can you think of using this gamma-globin cysteine synthetase with arsenite methyl transferase? So I think maybe it will be very useful for the crops.
So, thank you, Dr. Tripathi. Dr. Tripathi is basically saying that can be combined with the methyl transfer that he was showing yesterday. And almost like 7 years back, I called him, let's add this one on the top of my plants, and you do in your lab on the rice here. And it has not been done yet. So that was basically our idea that to go for that, good. But we haven't done, and we should do that.
Yeah, we tried to. Thanks.
Thank you.
Very nice talk, thank you. Just a quick question. How stable were your mutants of camelina? Genetically and phenotypically?
The mutants were Arabidopsis. The knockdown lines were Arabidopsis. Overexpression lines were camelina.
Yeah, how stable were those lines, genetically and physiologically?
Yeah, they are very stable. They are the tDNA lines. They have been tested and they are very stable.
Thank you.
Yeah, thank you.
Okay, thank you, Professor Om. And now we will call upon Professor Badara Sahib, Royal Military College of Canada, on phytoremediation of saline soils using halophytes. Professor Badara Sahib, online.
I think it's the version change. Professor Barbara Z will be presenting online. She has a recorded version, so she will be we will hear that. Oh, we are having some technical issue. It will be solved within some time. Like, can you just wait for 2 seconds?
Any questions? Any questions?
My co-authors are Dr. Lindsay Cartwright, a research scientist at Well, hello everyone at IPC 18. I'm not sure which session I ended up in, as there are no specific halophyte sessions at this conference, so likely this is a completely different talk than you were expecting. I work at the Royal Military College of Canada, which is a small federal university located in southeastern Canada. I'm going to speak today about phytoremediation of saline soils using halophytes. Specifically, I'm going to address the road salt issue in Canada. My co-authors are Dr. Lindsay Cartwright, a research scientist at the Toronto Region Conservation Authority, and 3 master's students, Lauren Nayroth, Amanda Chiasson, and Ellen Mann. Road salt is becoming a huge issue in Canada. As I said, my university, indicated by the yellow star, is located in the southern part of Canada, and even there we get harsh winters with significant snowfall and temperatures often exceeding -10 degrees Celsius in the winter. Due to our climate, road salt is liberally applied to roads between about November and March of each year to reduce the number of vehicle collisions. Because of this reliance on road salt, Canada is the #1 user of salt in the world, and salt usage is not decreasing in Canada, quite the opposite. Despite the Government of Canada's requirement for salt management plans, salt usage has increased from about 5 million tons per year in 2004 to about 7 million tons per year in 2021. The three main road de-icers we use are sodium chloride, magnesium chloride, and calcium chloride. While road salts are considered toxic under the Canadian Environmental Protection Act, there are no published guidelines that chloride must be maintained at in soils. Through a literature review of background chloride levels in Canada, we have established that anything over about 100 milligrams per kilogram or milligrams per liter is considered elevated, but we often try to get our sites even lower depending on the background soils at a particular site. This map of southern Canada highlights some hot spots for chloride in the summer months. So you can see concerningly high levels of chloride, up to 775 milligrams per kilogram, are routinely being observed even in the off-season. When snow melts on roads, sidewalks, and parking lots, all of which are heavily salted, chloride moves to stormwater ponds, groundwater, and soils where it is stored. Over time, this legacy chloride moves to freshwater streams and lakes. So even if we stopped salting completely, we would continue to see rising freshwater chloride levels. This is a problem as chloride levels in many freshwater sources are already above thresholds to protect aquatic life and human well-being. My research group has been working with halophytes for over a decade now. These salt-tolerant plants can be classified into three categories: excluders, accumulators, and excretors, based on their mechanism of dealing with salt ions. For the purposes of phytoremediation, we only use the accumulators and the excretors. I'll speak first about some of our accumulator halophyte projects, as accumulators are the most common type of halophyte. Their implementation is quite straightforward in that we plant them, we allow them to grow, we harvest them at the optimal time, and then we seek ways to repurpose the salty biomass. In this first study, we investigated the ability of three atriplex species, also known as saltbush, to accumulate road salt or sodium chloride from contaminated roadside soils. All three atriplex species are native or naturalized to southern Ontario and Canada. They all exhibit high sodium and chloride extraction rate capacities, and they are actually found quite often on disturbed roadsides already. So we carried out a greenhouse study with a parallel field study along a smaller highway running north of Kingston, Ontario, where my university is located. For the greenhouse study, we spiked the soil to levels that represented very high and high salt levels. The plants were grown in the spiked soil in my greenhouse for 2 months, after which the above ground plant material was harvested, separated into components, and analyzed for salt content. For the field study, we planted the three atriplex species adjacent to the secondary highway and harvested it after 4 months of growth. This graph displays the chloride concentrations in the above-ground plant tissue in the spiked and control soils. All three species, as you can see, were able to extract impressive amounts of salt. However, the only significant difference from the control was in Atriplex hortensis in the very high salt condition. This was because the controls were watered with tap water that actually contained a fair bit of salt, so even the control plants accumulated high amounts of salt. It was at this time that we determined that my university's water supply actually had extremely elevated levels of sodium chloride. We looked at the salt storage in the different components of the Atriplex species and determined that the leaves and the seeds, or the bracteoles, both held a lot of salt, indicating that harvesting all of the above ground shoots was optimal for all three species. Taking the extraction rates for the above ground tissue in each species and accounting for the optimal biomasses that we could achieve, we calculated that 2 of the 3 Atriplex species would take less than 10 years to remove all the salt from a heavily contaminated roadside. Now, typically roadside salt levels near Kingston are actually an order of magnitude lower than this scenario, meaning it would take much less time to remediate, but recall, we are also constantly adding more salt as we salt the roads each season. This study was important because it was the first to compare field and greenhouse salt extraction rates and to document the growth of the A triplex along roadsides. My group then carried out some mesocosm experiments to determine the amount of sodium and chloride in the soil profile after the spring melt and see how this was influenced by different salt application rates. We set up columns and filled them with soil to mimic a 20 centimeter soil profile with road salt application over the winter and observed the movement of salt through the soil after the spring melt. The soil profiles were split into 4 equal sections and analyzed for sodium in red and chloride in blue. And then we developed ion concentration gradients through the soil profile. A mass balance demonstrated that the majority of the sodium, or 87%, was retained in the soil through the winter season, with much of it retained in the upper part of the soil column. However, the chloride mostly leached out of the soil with only an 11% retention as it followed the movement of pore water. An analysis of the leachate collected confirmed this finding. So this stresses the importance of using appropriate halophytes to intercept excess chloride from runoff in the soil on an ongoing basis. I'm now going to show you one of our current studies that involves remediation of road salt in the Toronto area. The red box in this photo shows the location of the plot that we established in June of 2022. It's in an urban area where snow piling occurs in the winter and where spring runoff collects on its way to local water basins. During preliminary soil sampling, we established that the background chloride levels were 20 milligrams per kilogram, and there was up to 500 milligrams per kilogram chloride in the areas of highest contamination. So the goal of the remediation here is to reduce the chloride levels back to the site-specific background of 20 milligrams per kilogram. So here's a picture of the plot establishment, and we used seedlings here, purchased actually that were grown for us by a local nursery to ensure that the plants had a good chance of establishing. The 4 native species that we selected for the site consisted of side oats grama and prairie cordgrass being our excreter species, and switchgrass and sand-drop seed being our accumulators. Half of both accumulator plots were removed at the end of each season. However, the excreters don't need to be removed as they disperse the salt on their own and will grow back perennially. So here's the field site when we first established it. It consists of 5 plots, 1 for each of the species, plus a control to see what will grow opportunistically. The accumulators are shown here in white and the excreters in yellow. Due to the heterogeneous nature of the soil, remember our highest levels were at 500 by the time the site was rototilled, and we had a consistent level of 365 milligrams per kilogram chloride in the soil within the plots. And again, we're trying to remediate to the 20 milligram per kilogram background level. This is what the site looked like at the end of the first season, and you can see that all of the plants are doing quite well, with the switchgrass and prairie cordgrass performing the best. This was near the end of the second field season, and all of the plots except the sand-drop seed continued to thrive. And this was the situation at the end of the third field season, so only about a month ago. In early June of this year, the accumulator sand-drop seed was replaced with rough drop seed, which has done very well in its first season. As you can see from the data above, we have very good accumulation of chloride, particularly in the first and third year. 2023 was a challenging growing season in Canada with numerous cold spells. Most importantly, though, you can see the decrease in salt levels in the soil on the far right. Of course, some of this will reflect chloride washed out of the soil, but the chloride accumulation in the plant tissue on the left and the bioaccumulation factors shown at the bottom show that the halophytes are doing their job well. Taking into account the amount of chloride in the soil, the biomass production of each species, and the amount of chloride being taken up by the plants, we can calculate remediation timelines to get our soils to the background level of 20 milligrams per kilogram. And this is without taking into account the salt lost through the soil column. So these timelines shown in red are pretty good, with both the prairie cordgrass and the switchgrass remediating the site in under 3 years. The rough drop seed will likely perform well too, but as we only have a single year of data, we don't want to speculate on that one too much. Despite the timelines being very good, we're always being pushed to hasten the remediation timelines. Hence, we've started to work with bacterial symbionts that live within the halophyte plant tissues. These endophytes can promote plant growth and reduce plant stress, making them desirable candidates for enhancing phytoremediation efforts. This work is in progress now, and you can see a little more about it in the poster presented by my student Amanda Chiasson. We hope that the results of Amanda's study will provide valuable insights on the role of endophytes to salt phytoremediation and ultimately work to speed up the remediation process. So a good question that many people ask is, what do you do with the plant biomass? Essentially, any accumulator species used in phytoremediation does need to be harvested. We've harvested a lot of different plant species over the years, and what we routinely do with them is compost them. When we compost, we get 40 to 90% biomass reduction depending on the type of plant. So in this view here, I'm showing composting of the switchgrass. In fact, we also composted the sand drop seed. They were composted separately over a 9-month period and achieved 74% reduction in switchgrass biomass and about 60% reduction in sand drop seed biomass. After composting, the plant matter was saturated with water to remove the chloride present. 3 subsamples from each species were taken of the compost, about 20 grams each, were placed in distilled water, and the jars were shaken for 2 minutes and then left to settle for 2 days, and the rinse water was collected. This process was repeated 3 times. The chloride was definitely reduced significantly with this composting and rinsing regime. We did do electrical conductivity measurements after each rinse, and it indicated quite a steady decrease in chloride. So overall, 99% of the chloride was removed during this rinsing process. However, as you can see, this was done on a very small scale, and we now need to optimize the process and seek ways to make it more viable for large-scale operations. And that work is currently in progress. I do want to briefly mention our work with Recreto halophytes. Only a small number of halophytes are able to secrete salts from their leaves. There's about 370 Recreto halophyte species worldwide, and they are distributed widely. We have 15 native Canadian Recreto halophytes. You can see the salt accumulation here on one of them, Disticulus spicata, or an inland saltgrass. And with the aid of the scanning electron microscope on the right, you can see the salt crystals on the leaves and even the specialized salt glands on the leaf surfaces on the bottom right. So the idea of Recreto halophytes is to get this process of haloconduction working. So in the Recreto halophytes, just like in the accumulator halophytes, salts are taken up by the roots with the soil water, which is then translocated to the shoots via the xylem. Rather than being sequestered in the vacuoles, the salts are transferred to specialized salt glands on the leaf surfaces, which secrete the salts in the form of a concentrated liquid droplet. As the droplet dries, the salts crystallize out, as you can see in the little inset here. And here's a picture of how we can get a nice billowy amount of salt sort of accumulating on the outside of the plants. Now, when the wind blows, you get movement of the salt particles into the air column. The salt particles will become diluted with increasing distance from the source and will eventually be deposited on the soil at a distance from the site. Most Recreto halophytes will excrete measurable levels of salts in their second year of growth, and these plants never need to be harvested, and so this labor-intensive step is eliminated. We have created models to show that under appropriate conditions of humidity and wind, a good percentage of the excreted salt can be widely dispersed and deposited at distances of at least 5 kilometers and probably more at levels well below background, at which point the salt ions act as plant nutrients rather than contaminants. So even though we don't usually like to do it, dilution is definitely a good solution when you're talking about salt remediation. So where does this leave us? Well, basically, it tells me that phytotechnologies can and should play a role in salt remediation strategies, and particularly in road salt remediation strategies in Canada. It is very important to intercept road salts before they enter freshwater sources. We know the chloride ions move quickly through the soil column following the movement of pore water, and we know that native halophytes have the ability to remove significant levels of salt from contaminated roadsides. We now need to seek efficient ways in which to harvest the accumulator halophytes and to repurpose the salty biomass. To that end, we have partnerships with the Ministry of Transportation along our major highways, with the 407 ETR, which is a major bypass highway for the Greater Toronto Area, and with the Pearson Airport Authority. And all of this is bringing us closer to increasing regulatory approval. I thank you all for listening, and if there is any time, and if I have managed to get online, I would be very pleased to answer any of your questions. Thank you.
So thank you very much. With this, we are concluding this session. On climate change and carbon sequestration. She's on.
Give the time to questions for Barbara.
Hello, Barbara. Are you there?
It was pre-recorded though.
Yeah, I didn't know she's there.
She's not online. She's not online.
She's not online.
No, she's
not.
No, she's not.
Okay.
We cannot ask you. It's not live. It's not live.
Yeah. Please put off that speaker.
Amrita, stop share first. Stop share on your computer. And because speakers are on. So let's have coffee.
Yeah. So.
Here on time, please. 11:05.
Two speakers and 3 student speaker who will be judged for their presentations, and on the basis of that they will be rewarded. But first we'll start with our first speaker after tea session. He is Shirish Srivastava from ICGB New Delhi, and he will be talking about improving growth and carbohydrate production in a marine cyanobacterium. Dr. Shirish, please.
Good morning, everyone. Am I audible now? All right. So, first of all, thanks to the organizers for giving me an opportunity to come here, and also thanks to the session chairs for letting me talk today. I'm Shirish Srivastav. I'm Group Leader of Systems Biology for Biofuel Group at the International Center for Genetic Engineering and Biotechnology. One of the major interests of our group is to understand the metabolism of primarily cyanobacterium cells for production of biofuels and bioproducts photosynthetically. So today what I'll be talking about is one of our works where we have improved the growth and carbohydrate production in a marine strain, cyanobacterium strain, through overexpression of carbon transporters, and we have also evaluated the effect of different promoters and RBS on the overexpression of these carbonate transporters. So I come from ICGB, which is an international organization in the UN system, which means it has 68 member states, which are different nations, which are members of this institute, and there are 18 signatory states whose membership is pending, and we primarily have 3 host countries. One, the primary headquarters is in Trieste, a major component is in New Delhi, and another component is in Cape Town, South Africa. So we do a lot of research in our labs at all of these 3 components, but we also support both advanced education and training through long and short-term fellowships for PhD students and postdoctoral fellows. We organize many meetings and courses and workshops to train scientific personnel in these areas of biotechnology. And we also offer a lot of competitive research grants for supporting earlier career research scientists. We do technology transfer to industry for production of biotherapeutics and diagnostics, and we are involved in biosafety, bioethics, and other institutional activities. So I come from ICGB New Delhi. Website of ICGB can be accessed here, as well as information about meetings and courses can be accessed here. So a lot of these meetings are international in nature, where people from different countries come over and stay with us for 10 to 15 days and attend various hands-on trainings and courses. So coming back to the scientific problem, we all are very well aware of the global carbon dioxide emission problem, and as previous speakers have also mentioned, that the global greenhouse gas emissions have been increasing at almost an exponential growth, and correspondingly the global temperature anomaly has also been increasing. And in terms of country-wise greenhouse gas emissions, if you look, the primary culprits are all the developing and developed nations, for example, US, Canada, India, China, all of these, Brazil, these are all the countries which contribute most to this CO2 emission problem. And of course, keeping the temperature anomaly to less than 105 degrees Celsius is very, very essential. So we are running against time, because otherwise we are just knocking the door of global catastrophe. So we require to develop biological organisms which can efficiently capture carbon and then can be utilized for various applications. And here I'm proposing cyanobacteria as an efficient carbon capturing biocatalyst. So the reason is that they are the fastest growing photoautotrophs with doubling time of about 2.5 hours for the best strains. They have very minimal nutrient requirements. Especially the marine strains do not require fresh water, so of course there is always a competition between fresh water and the marine things, and India itself has a 7,500 kilometer coastline, so we are well set to utilize marine strains. They store carbon as glycogen, which is a glucose polymer, which can then be hydrolyzed and used in various applications. And under suitable conditions, the glycogen levels in these cells can go as high as 45 to 50% of their biomass. and the remaining biomass, a major fraction, is effectively proteins. And for a lot of other biofuel and biochemical sort of applications, compared to agricultural biomass, especially for the unicellular cyanobacteria, we do not have any lignin, so the hydrolysis is much easier, and we get primarily glucose as our sugar. And at least theoretically, it's likely that the productivity of these cyanobacterial crops is much higher than even those of energy crops. Last but not the least, these are naturally transformable and have high rates of homologous recombination, which means it's much easy to genetically engineer them to make different products. So for this work, we have specially focused on Synechococcus 7002 cells, which are a fast-growing unicellular marine cyanobacterium, which has reported doubling time of around 2.5 hours under ideal conditions. Another advantage of this strain is its high optimum temperature of around 38 degrees Celsius. So for warmer climate like that of India, this is very important. And they are also resistant to high elimination. And from the genetic engineering perspective, their information about it, all the genes as well as their neutral sites is known. Their general limitations are that typically they have low glycogen levels, about 15 to 17% under normal growth conditions. If you really want to have high growth rates, so in terms of high carbon capture, you require more than 1% CO2 bubbling to achieve faster growth rates. So which obviously indicates that the growth and the carbon capture is limited by carbon transport and can be increased by increasing the carbon transport. So overall, our idea was, can we utilize, so in terms of carbon capture, there are two modalities. One is direct air capture or DAC, and the other one is you concentrate the CO2 and capture it in some chemical medium and then release it and have a concentrated stream from which you can capture further. So especially for cyanobacterial work and biocatalyst, we want to aim towards capturing directly from air, which is lower carbon content. So that's why we went for engineering these strains so that we can directly capture carbon from air and have higher biomass growth rate. In terms of carbon transporters in cyanobacteria, there are different kinds of carbon transporters. Cyanobacteria also have these unique carbon concentrating mechanism with the presence of these carboxysomes, which basically make the carbon capturing more efficient. The primary carbon transporters which are on the surface of the cells are mainly you have CO2 transporters as well as a series of bicarbonate transporters. We have focused specifically on bicarbonate transporters for this work because under the alkaline pH conditions where we grow these cultures, bicarbonate is the primary carbon dioxide source species. And within those, we have specifically looked at SBTA or sodium dependent bicarbonate transporter, which is a high affinity inducible bicarbonate transporter, as well as BK, which is a lower affinity and high flux constitutive transporter. Also for BK, a previous study has shown that its expression in 6803 cells, which is freshwater strain, can improve their growth. So what we wanted to test was compare these two transporter and their overexpression and their impact on growth and carbon capture in terms of glycogen. So general construct design is we have these flanking regions with neutral sites. Neutral sites are those sites where if you put in any genetic element, it does not impact the physiological response of the cells. We have promoters, RBS, the target gene, antibiotic resistance gene for selection, and a terminator. And as I have mentioned, we have utilized natural transformation for our transforming the cells, followed by antibiotic selection. The transformants were verified by PCR. So in terms of design elements of all these components, we have tested different combinations. We have two target genes, SBTA primarily and BK secondarily. We tested different promoters, RBCL2A, CPCB, and CPCB560, and two different RBS combinations. So for the first set of work, we have tested the comparative effect of SBTA versus BK overexpression. So the construct were almost identical. Both of these genes were expressed under RBCL2A, and the relative expression of these genes compared to the wild type was measured through RT-PCR. Now, for SBTA, which has very low basal level of expression, we have seen about 70-fold increase in the mRNA of this gene, while for BK, which is constitutive and has a reasonable expression, the relative change versus wild type was lower, but still it was around 15 to 17-fold. Now, when we grew these cultures in presence of air, both these transformants, A stands for cells expressing SBTA, B stands for cells expressing BK. Both of these grew to almost double the rates of the wild type cells. And we have, though I have not shown here, we have also cultured these cells in elevated CO2, for example, 1% CO2 bubbling as well as 3% CO2 bubbling. And what we have seen is that even in higher CO2 conditions, there was increased biomass productivity, which is defined as how much biomass we can get in 1 liter of a culture in a day. So wild type cells on air produced roughly around 100 milligram per liter per day, while the transformants could go up to 200 on air, or even up to 270, 260, 270 with 3% CO2. And what we have also seen was, in terms of lower CO2 concentrations, both BK as well as SBTA were comparable, but when you go for higher CO2 concentrations, BK was slightly better than SBTA. We also found that the glycogen content of these transformants was significantly increased. So from basal level of around 20%, it went up to 34% for air-grown cultures. Even for CO2-grown cultures, it was higher, though the increase was not as appreciable, even though statistically significant as it was for the air-grown cultures. So in terms of glycogen productivity, or how much glycogen you can make per liter of culture per day, the productivity was increased twofold. That means it became 3 times. So from roughly around 20 milligram per liter per day, it went up to 60 milligram per liter per day on air-grown cells, and similarly higher productivities, even up to 80 milligram per liter per day, were seen in the transformants. So we were able to increase the glycogen levels in the cells to 70% and 200% increase in the glycogen productivity. So encouraged with these results, we then tested different RBS and promoter combinations, in specifically focusing this time on SBTA overexpression, because there have not been any studies on SBTA overexpression in these cells. And for this, we tested 3 different promoters, as I have mentioned previously: CPCB, CPCB560, and RBCL2A, and 2 different RBS combinations. The construct design was very similar to the previous time. The only thing is what we changed was the promoters. And again, we tested the positive transformants through amplification of the neutral site regions, with PCR. The relative expression of these genes were tested. In this case, SBTA was tested through RT-PCR, and what we have seen was, we have seen increase of anywhere from 60-fold to up to 90-fold increase in SBTA overexpression, which was also verified through Western blot. And what we have seen was, again, there was a dramatic increase in biomass as well as glycogen content of these cells in with both air bubbling as well as CO2 bubbling, where the glycogen content went up to again about 35-38% in the transformed cells, and the biomass productivity also went higher. So here we could get about 80 milligram per liter per day of productivity with air. Now again, it is also very important that the biomass productivities as well as the glycogen productivities, what we are seeing in transformants with air culture is comparable to what we are seeing for the wild type cultures, or even higher than what we are seeing with CO2 bubbling. So this shows that these transformants are able to sequester more CO2 and make more of glycogen.
Shirish, okay.
Yeah, all right. So I guess you guys got the idea. So thank you.
Yeah, it's excellent presentation. Thank you. Now it's open for taking 1-2 questions. I have it. Sure. Yeah. So, what's the carbon fixation mechanisms here that its fixation is more as compared to the other angiosperms or other algae, or what is their main?
Right. So in this particular case, what we have done is we have put in more transporters. So the transport of CO2 is increased. And what we already know is that these have these special carboxysomes which are very efficient in terms of their CCM or carbon capturing mechanism. So because of this combination of these two, plus the third factor is purely physical. You have cell size which is individual cells which are much smaller, so the carbon transport can occur from all directions.
Okay, thank you.
How was the nitrogen fixing ability of this cyanobacterium?
No, these are not nitrogen fixing cells. So we had supplied nitrate in the medium.
Nitrate in the form of ammonium or potassium?
Sodium nitrate.
Sodium.
Yes.
For carbon fixation, sir, can I ask? Yeah. For carbon fixation, do we need the transporter? Generally, you have carbonic anhydrases in algae. They can do the carbon fixation, right?
No. So for base, as I've seen, the basal growth rate is there. If you want to have higher carbon transport, if you want to grow more cells, if you want to fix more carbon, then of course, as what we have shown here is that putting more transporters increases.
But carbon can diffuse. Why you need a transporter there?
No, so under pH conditions of basic, it's primarily bicarbonate, which is a charged molecule. So it cannot diffuse.
Any other questions? Okay. Thank you, Shirish. Thank you.
Thank you.
Our next speaker is Kalipara Padmasri. She prize for this presentation, so they should strictly follow the guidelines they have been given for the presentation so that they can earn more point on those issues. And the first presentation, so should I spell out the guidelines again, or the students, I mean it is fine. So total presentation time is 15 minutes, 12 minutes for the presentation, and 3 minutes for question and answers. So first student is Mr. Joel Joy. He is from University of Calicut, and he will be talking about herbascular mycorrhizal fungi, an apt microbial biostimulant for enhancing photosynthesis and salinity stress tolerance in rice. Mr. Joel, your time start now.
Good morning all. So as said, the topic of my presentation is herbascular mycorrhizal fungi, an apt microbial biostimulant for enhancing photosynthesis and salinity stress tolerance in rice. Coming to the introductory part, the global climate change as well as the increasing human populations are two increasing or biggest challenges that the current human population is facing. And because of the global climate change, various environmental stress factors are directly hampering the growth of these crop plants. In my study, it's the rice plant, and I have chosen salinity stress as my primary objective of the study. When we see the rice production worldwide, it has significantly reduced from 751 million tons in 2016 to 695 million tons in the year 2018. And according to the Food and Agricultural Organization in the year 2018 reports, there have been increasing trends that the amount of rice production will steadily decrease as the year goes past by. So we need to develop strategies to protect the rice production. Rice is a crop which is actually extremely sensitive to salt stress. When the salt stress levels are applied beyond its threshold level. So my present study is to find out the protective mechanisms of the AMF under NaCl stress condition through controlled pot experiment. Now coming to the methodology, materials and methods part. The seeds for the experiment were bought from KU Akshaya, Kerala Agricultural University. They were properly sterilized and then they were allowed to germinate in the soil which were initially colonized with the mycorrhizal inoculum. So prior to the growth of these seeds, we need to develop a suitable host to develop the sufficient amount of inoculum. So the suitable host which we used is maize plant. So the speciality of these root mycorrhizal fungi is that they colonize the root cortical cells and then they ramify within the root cortical cells, develop arbuscules, structures known as arbuscules and vesicles. Arbuscules serve as the nutrient exchange points in these cells. So these structures help in acquisition of various nutrients inside the plant, especially when they are not available to the plant as such. So the NaCl stress condition where then initially these rice seeds were then allowed for germinating in these soil conditions, and upon reaching the 35th day, the salt stress conditions were imparted by applying salt solution thrice, and on the 55th day of the growth, various photosynthetic responses were recorded. Parameters such as pigment composition, MDA content, hydrogen peroxide content, superoxide content were analyzed. Various photosynthetic parameters with the help of handy P, photosynthetic plant photo efficiency analyzer, were used for determining the quantum efficiency, maximum quantum yield, efficiency of water splitting component has actually colonized within the cells. To verify whether our mycorrhizae has actually infected or colonized the root cells, we will stain with a specific stain, trypan blue, to see the various structures. Here you can see arbuscules and vesicles within the root cortical cells. Now that the mycorrhizal inoculum has been properly ramified within the root cortical cells, the soil proteins and proteases were analyzed. Here the total GRSP content, the significance of GRSP is that these glomalin proteins is an exact indicator of these mycorrhizal fungal inoculums. So as we can see, there are 4 treatments as said. The control C stands for the control, C plus M stands for the control plus mycorrhizal treatment, S stands for the stressed conditions, S plus M stands for the stress plus mycorrhizal treatment. The total GRSP content was found to be the highest in control plus mycorrhizal treatment, which was then followed by stress plus mycorrhizal treatment. This trend was seen among the easily extractable and difficultly extractable GRSP content. Moreover, we have also checked the soil protease activity. This is because protease activity has significance in recycling of the organic nutrients. So if the amount of soil protease is the highest, then the complex protein could be easily recycled into shorter form and made available to the plant as such. The chlorophyll pigment such as carotenoid, total chlorophyll, chlorophyll A, chlorophyll B were also analyzed, and among all a similar trend was seen. Control plus mycorrhizal treatment was found to be the highest for all the treatments. To understand the to understand the membrane damage, hydrogen peroxide and superoxide as well as MDA content was seen, and among them the stress plus mycorrhizal treatment were found to be generally the highest. Now to see whether this has it any benefits for the chlorophyll or the photosynthetic apparatus, various photosynthetic parameters were analyzed using HandyP. Initially the FO, initial fluorescence, and FM, the maximal fluorescence data was recorded, and as it can be seen, the stress condition had the highest amount of initial fluorescence, whereas the maximal fluorescence had the stress conditions. The FO by FM ratio was found to be the highest for stress conditions. The water splitting complex, the FV by FO ratio, was the least for the stress condition, indicating that the efficiency of photosystem 2 in splitting the water splitting complex has been damaged exteriorly. The photosynthetic efficiency, quantum yield efficiency, PO, was found to be the least for stress conditions, which are followed by stress plus mycorrhizal treatment. Absorptive flux per reaction center was found to have the least for the stress condition, followed by the control plus mycorrhizal treatment. The trapping flux plus per reaction center was also found to be the least for the stress condition, again indicating decreased efficiency of photosynthesis. The performance index was also found to be the least for the stress conditions. Similar was the case of structure to function index, where the stress condition was found to be the least. Coming to the conclusion, out of all the microbial biostimulants, we have developed a mycorrhizal inoculum which could counter protect these damages caused by the salt stress, and they could also modulate the soil enzyme. I kindly acknowledge the University Grants Commission as well as University of Calicut for providing me an opportunity to present. Thank you.
Thank you, Joel. Now it is open for questions.
Yeah. Joel, it's a good presentation. Just one clarification. How specific this mycorrhizal for the salt stress?
So how specific is?
For the protection of all the photosynthetic parameters, how specific? Can it act against temperature or any other stress?
Yeah, actually mycorrhizal inoculum, these fungi already has many number of benefits, but we have actually studied only for salt stress. They have already established heavy metal tolerance, imparting property to the crop plants.
Mm-hmm. No, generally salt is a different mechanism because create an osmotic mechanism. Yeah. Temperature creates a different mechanism.
So actually mycorrhizal herbuscules which are developed within the root cortical cells, they help in exchanging of these nutrients. So they actually help in competing of the Na plus as well as Cl minus ions, thereby preventing its entry into the plant cells. Instead, they will help to transport the essential nutrients from the soil.
Good. Yeah. Yes.
The mycorrhiza which you have, is it halotolerant?
No, it is actually, we are yet to find out. We are in the process of finding it.
So you don't know the species you are working of that?
Species.
Right now it is not confirmed.
Yeah, the species is there, but studies on halo tolerant have not been done.
Just one basic question.
Yes.
So when it is giving the mycorrhizal inoculation, it is giving a better effect on plant growth.
Yes.
Is it like promoting the exclusion of sodium ion from roots, or it is sequestering it somewhere, or just by
No, excluding the sodium ions from or preventing its entry from the soil into the roots.
Yeah, okay. But you don't have that data to support right now.
One question, yeah, please.
No, it was on similar lines with Sir
Then
has
fine,
asked.
then fine.
Yeah.
Yeah.
Actually, we have done an ICP-MS analysis, but I have not kept the data in the presentation to verify those results.
Just a query. What is the duration of the variety?
Variety, the entire duration, it's 4 months.
4 months.
4 months.
And you have picked 55 days as the. What is the reason?
Reason is actually the proper colonization takes 45 to 50 days for 100% colonization, because the seedling establishment and the mycorrhizal growth together needs 45 to 50 days to fully colonize.
But maximum photosynthesis will accumulate at what stage? It is only the bacterial, fungal, or the growth of the plant is also essential?
No, both are essential. But when we see the benefits of the mycorrhizae, it's important to see like its growth has to be occur till 100%.
Okay, if there is no any more questions. Oh, okay, go ahead.
Thank you for your presentation. I have some basic question about methodology. How many replicates did you have?
3 replicates.
3 replicates per treatment, right?
Yeah.
And the concentration of salt, maybe you mentioned, I missed that. What concentration did you use?
I have used 100 millimolar of NaCl.
Thank you.
For the stress imparting concentration.
Thank you.
Yeah, just to clarify, the standard deviation that we see in all the experiment, I see standard deviation is perfectly balanced. Please check your data again.
So actually, okay, I will check, sir.
Okay, thanks, Joy. So our next talk by the student only. So Priyanka Prajapati from Banaras Hindu University, Varanasi. Endophytic fungi accelerate the ascorbate glutathione of wheat in response to the salt stress. So your time for 12 minutes for your presentation and 3 minutes for the questioning session. Okay, Priyanka, now your time starts.
Good afternoon, everyone. So I'm here to present my work, Endophytic fungi accelerate ascorbate glutathione of wheat in response to salt stress. So why the abiotic stresses? Because we know that the current population of the world is 8 billion, which is in next 50 years will become the 9.7 billion. So this increasing human population became the global problem. So to feed this human population, huge population, we need more food. So day by day, the crops are being affected by abiotic stresses like salinity, drought, temperature, cold, etc. So how do we get the more food to feed the increasing population? So there's the different approaches, increasing agricultural land, greater use of the chemicals, safe and efficient pesticide, more farm mechanization, greater use of the transgenic crops, and the expanded use of the fungal endophyte. So except the expanded use of the fungal endophyte, the other approaches became failed because of the limited resources against the eco-friendly approach, and restricted the greater use of the transgenic crops restricted due to the ethical concern. So the endophyte, expanded use of the fungal endophyte, yes, because it is eco-friendly and it is very cheaper. And we know the fungal endophyte protect the plant from abiotic stresses by secreting some natural compound. So yes, we can, by using the fungal endophyte, we can enhance the food to solve the problem of the food security. Introduction. So wheat is one of the world most essential cereal crops, providing a significant portion of the global food supply and serving as a staple for millions of people. However, its productivity is severely affected by various environmental stresses, among which the salinity is the major abiotic stress, which affect the plant growth, leading to reduced yield and economic losses in agriculture system worldwide. So it became a very urgent imperative to develop the sustainable strategy to enhance the crop tolerance and improve the agricultural productivity. The term endophyte. The first time the endophyte term was given by Antoine Dubarry in 1866. He defined the endophyte are the microorganisms. They are present within the living healthy tissue of the plant without causing any disease. Plant growth promoting fungi. They are the heterogeneous group of non-pathogenic saprotroph fungi and the exchange metabolite with the plant directly. So they have also the beneficial effect on suppressing the phytopathogenic microorganism and also they playing very important role in regulating plant responses to abiotic stress by inducing stress related gene and modulating antioxidant defense system. Despite a growing body of research on the beneficial effect of endophytic fungi in enhancing the plant development and stress tolerance, there is still a knowledge gap regarding their specific role in promoting wheat growth and conferring salt tolerance. So the present study aimed to address the gap by investigating the effect of selected fungal endophyte on wheat plant grown under saline condition. So we hypothesized that the endophyte could improve the wheat growth and enhance the salt tolerance by positively modulating the ascorbate glutathione cycle and regulating stress responsive gene expression. So by addressing this gap and hypothesis, we following objective were designed. So in our pilot experiment, we procured the two different variety of the wheat seed salt tolerant, that is the KRL 213 19-10 from the CSSRI Haryana and salt sensitive variety of the wheat seed from Agricultural Institute of the BHU, that is the PBW 343. So they were the screen at the different concentration of the NaCl up to 300 millimole. The EC50 value was found for the salt tolerant variety 250 millimole and for the salt sensitive 100 millimole. So by selecting this zone, the wheat seedling was grown up to 21 days and the endophytic fungi were isolated from the different tissue of the plant, and they were further approx 50 endophytic fungi were isolated from the both variety, and the variety and the fungal endophyte which were found common in both variety, they were discarded, and the unique endophytic fungi, they were further screened up to 1 molar NaCl. So out of the 50, 11 were found salt tolerant, and they were further screened from the biochemical assay, PGP of extracellular enzyme activities and the antifungal. So out of the 11, the 4 were found they gave the positive result for all biochemical assay. So for the consortium application, they were further selected for the synergistic and antagonistic effect. So out of the 4, 2 fungus gave the synergistic effect to check their consortium application, the pot experiment. So these 2 fungus were molecularly identified, that is the Cladosporium parahalotolerant and Aspergillus medius. Okay. Now, in the present study, this work has been published in Microbiological Research. So, now in the present study, I am going to discuss about how the spore suspension preparation of these 2 fungus, and after that, how the 8 treatment were made: control, NaCl, first fungus. Fourth is first fungus plus stress, and the 5th is second fungus, that is Aspergillus medius. Sixth is Aspergillus medius plus stress. 7th is consortium, and at the consortium plus stress, that is the 100 millimole NaCl. So now they were further checked for the following parameter per study, that is the measurement of the fresh and dry weight of the root and shoot, measurement of the MDA level, soluble sugar total chlorophyll and protein content, assay of the antioxidant metabolite, assay of the antioxidant enzyme activities, extraction of the total RNA and analysis of the gene expression by quantitative real-time PCR, and also the quantification of the photosynthetic pigment were observed. So in the first objective, I'm going to discuss about the in vivo study on interaction of the Triticum with infestation of the selected fungal endophyte, and to study the physiological and biochemical trait of endophyte treated salt sensitive wheat genotype under the salt stress. So result and discussion. So we can see the salt stress dramatically decrease the fresh and dry weight of the root and shoot. We can see from the picture and table. But the consortium application significantly enhance the root and shoot dry weight of the fresh and dry weight of the shoot and root. This result indicating they act as the plant growth booster and also act as the plant catalyst in the agriculture. MDA estimation. So MDA is the indicator of the oxidative stress which cause the damage of the cell membrane in the plant due to the excessive production of the free radicals. So the plant treated with the NaCl, they dramatically considerably enhance the MDA content. So this but the plant treated with the plant treated with the consortium, they significantly decrease. So it means in order to protect the plant from the oxidative damage. Photosynthetic pigment estimation. This result also showing the NaCl, they significantly decrease the chlorophyll A, chlorophyll B and total chlorophyll content as compared to the other treatment. But the consortium application with and without stress condition, they significantly enhance the chlorophyll A, chlorophyll B and net chlorophyll content. So this is showing that under stress condition, they enhance the decrease the pigment photooxidation and restore the chloroplast pigment activity. This table showing the same result in the percentage. The consortium application enhance the chlorophyll. What's happening? Some slide are not visible. Effect of the potential fungal endophyte and the carbohydrate, protein and reducing sugar content. So this result showing the consortium application significantly enhance the carbohydrate, protein and reducing sugar under the stress condition, which is ranged from the 70.27 to 71 in the consortium application, and under the stress condition it enhanced from the 50.98 to 44.6%. So these all act as the osmoprotectant which protect the plant from the oxidative stress under the stress condition. In objective 2, I am going to discuss about the study of the ascorbate glutathione cycle enzymatic and non-enzymatic component and the evaluation of the endophyte modulated gene expression under the salt stress by quantitative real-time reverse transcriptase PCR. Exploration of the ascorbate glutathione cycle. This is very efficient and detoxifying process under the stress condition which protect the plant from oxidative stress. So this cycle contains the enzymatic, non-enzymatic component. The enzymatic component and the change pattern of the glutathione oxidase. This table showing the change pattern of the glutathione oxidase, glutathione ascorbate. So the salt stress, they decrease the ascorbate and glutathione content but the consortium application significantly enhance in order to protect the plant from the oxidative stress. And in the stress plant, they take up the very sizable amount of the glutathione because to maintain its redox homeostasis. The assay of the antioxidant enzyme activities. So the 5 enzyme were observed the activity of the 5 enzyme SOD, APX, THAR, MADHAR and GR. So the consortium application significantly enhanced all enzymatic activity as compared to the other treatment, showing because under the stress condition this intensify the cellular defense mechanism to protect the plant from the oxidative stress. And this table showing the percentage enhancement in the all enzymatic activities, which is range from 32.27 to 91.91%. So this is the this is the principal component analysis. This is done due to find out the antioxidant variation in relation to endophyte inoculation as well as the salt intensity. So this we can see in the principal component P1 describe about the 66.04% variation, and whether the P2 described about 18.20. So all the enzymatic activities belong to principal component 1 except the MDA and oxidized glutathione. This table showing the DNA sequence of the PCR primer that we used for the qPCR determination of the 5 ascorbate glutathione biosynthesis related gene. So this table describes the gene accession number, primer pairs, and expected amplification size. Gene expression analysis by real-time PCR. So we can see in the SOD and APX, under the stress condition, this activity enhanced. So this showing there's a positive correlation between these two enzymes. But further, the consortium application significantly enhanced upregulated the transcript level. This is because of the overexpression of the transcription.
Okay, Priyanka, trying to finish it.
And translation, this is the silent finding of the study. So it was concluded that our research Aspergillus medes and Cladosporium parahalotolerant, they have remarkably ability to mitigate the detrimental effect of salt stress on the development and growth of wheat seedling. Further comprehensive investigation are required to ascertain which substances.
Your time is over. Just quickly, just 1 or 2 line conclusion.
Thank you so much for your attention.
Good. So Priyanka, very good presentation. Now the session is open for the questions. 1 or 2 quick questions.
So you said that your lab identified these endophytes from different parts of the plants. So these 2 endophytes, where they stay in the plant? Root, shoot, which organ? Root. Surprisingly, you did not show any colonization.
Sir, I have explained in the pilot experiment I have done. Actually, sir.
Yeah, so did you
I didn't
check
tell, but I have done, yes, sir, in previous study.
Not previous, but every experiment you should look into the colonization data also. How
sir,
much
I have
is
done the root colonization essay. This slide.
Where? Achha, okay.
Yeah,
It's
sir.
smaller.
Throw same and fluorescent to check whether they are colonized the surface and internal tissue of the
So
plant.
the colonization was uniform in all the plants?
Yes, sir.
Hello. Have you checked the yield data also?
No, sir, it is under the process. Actually, some of my slides are missing in this PPT, so I couldn't.
Then how can say that
Sir,
is the
this
tolerant
experiment
plant?
is
They
still
may be
going
the plant using here avoidance mechanism. Then how
Sorry,
can say
sir,
this is the tolerant plants?
Sorry, sir. Where? Oh.
Priyanka, he is here.
My question
Okay,
is that have you checked the yield data?
Sir, this experiment is still going on. Actually, I put the slide in PPT, but this is not showing.
Priyanka, first listen the question, then try to answer. Okay.
Have you checked the yield of the plants also?
No, sir, not yet. But, sir, I am doing that experiment in the field trial. It's still going on.
Okay. How you maintain the salinity in pots? Which method you have used?
Sanity. Sir, how I give the treatment to the
Yeah.
Sir, I did in the gradual process lab 25 millimole gradually to avoid the salinity shock. I didn't give the treatment of directly 100 millimole.
It is
So,
through seed
25
sowing and you mix the salt with soil.
Yes, sir. First, I mix the soil with the 25 millimole of NaCl gradually and then maintain the 100 millimole NaCl to avoid the salinity shock.
And have you checked the EC regularly?
Yes, sir. Yes, sir.
They maintain.
Yes, sir.
Oh.
Okay, any more question? No. Okay, thank you, Priyanka. So Again, next presentation by the student Noble Louis, University of Calicut, Kerala. UV radiation priming as a novel tool for manifesting the viability of rice seed without entering into the thermal dormancy. Okay, Luis. Now your time starts.
Good afternoon. And my topic is on UVB radiation priming as a novel tool for maintaining viability of rice seeds without entering into thermal dormancy. So, as you all know that the abiotic stress limits the plant growth and the yield. And the population is getting high and we need to produce more. And because of these abiotic factors, it limits the productivity of all crops. Especially in the case of heat, every 1 degree rise in the global level will cause to 5 to 15% of drop in the crop production. So in the case of germination, we have 3 phases. There is phase 1, phase 2 and phase 3. The phase 1 is the imbibition phase, phase 2 is the metabolic phase, and phase 3 is the germination phase. So in the case of Rasa sativa, like in the variety which I took, the phase 2 starts at 20th hour, and when I applied heat stress at the phase 2, this is what I got. The heat stress is 45 degree Celsius for 9 hours, and you can see the control plants. Then we have 14 hours, 16 hours. 14 hours means I give the stress that is 45 degrees Celsius for 9 hours at the 40 hours after the imbibition. Then 16 hours, this 14 and 16 represent the later imbibition stages, and the 18 and 20 represent the early stages of phase 2, that is the metabolic phase, and the 22nd and 24 represent the continuation of the phase 2. So from this picture it is clear that the plant struggles, that is the early stage of phase 2, that is 18 hours and 20 hours of the germination, is the most sensitive towards the heat stress in the rice seeds. And in this study, I have used different intensities of heat stress, that is 35 degrees Celsius, 40 degrees Celsius, 45 degrees Celsius, and 50 degrees Celsius. And the time and the duration is 9 hours. And it is clearly seen that the germination rate and the seedling survival rate is getting decreased. And for my study, I took 45 degrees Celsius for 9 hours at the 20th hour after imbibition for my study. So this is the scenario. When we apply the heat stress, the germination rate and seed surviving rate is getting down. So for that, we applied some priming techniques. That is, as you all know that the priming is kind of green vaccination. That is, we are applying a mild amount of stress to the plants, which using different priming agents, and which can increase the plant stress tolerance. And there is 2 types of priming. One is seed priming and another is seedling priming. And in my study, I go for the seed priming. And my seed priming agent is UVB priming, and the dosage is standardized and fixed, that is 12 kilojoule per meter square. So coming again, that is the phase 1, phase 2 and phase 3. So the priming, that is the UVB, is applied to the seeds during the 20th hours after the imbibition. Then after, the heat stress is applied. Then this is the result. So in the case of germination rate, you can see that yellow line indicate the heat stress alone, and when we applied the gray line indicate the heat stress, and when we apply the UVB, the germination rate get increased. That is the yellow line. And the similar result you can see in the case of seedling survival, seedling vigor, and the weighted germination index. In all this, the UVB treated plants are performing better than the heat stressed ones. And these are the phenotypes. Here we have the control. Then next we have the UVB. Then we have heat stress, and next we have heat stress plus UVB. So it is clear that like you can see how the seeds are struggling to germinate and how they are struggling to get established in the case of heat stress alone. And when I applied the UVB, you can see an increase. The seeds are performing better, showing higher shoot length, root length, and seedling dry weight and fresh weight, etc. Then moving to the total chlorophyll content and chlorophyll stability index and total carotenoid. In all this, it is shown that there is a hike in the plants which are showing higher performance is that UVB treated plants. Then we have lipid peroxidation, ROS, and electrolyte leakage. So first we have MDA. When the stress is more, the plant will produce more MDA. So in the case of the red one is heat stress, it produces more MDA. But when the priming is applied, the amount of MDA is reduced. And this MDA is produced because of the activity of ROS. That here we have hydrogen peroxide and superoxide. And it is clear that in both of these superoxide ROS are reduced in the case of UVB primed plants. Because of this ROS, it will causes the electrolytic leakage. And there is, you can see in the case of electrolytic leakage, heat stress have more ROS and more electrolytic leakage. But when the UVB is applied, the electrolytic leakage gone down. And in the case of membrane stability index, if the plant is more stressed, more ROS, and the membrane stability index will go down. So that result you can see in the case of heat stress plants, and better stability is shown by the UVB primed plants. And in the case of anthocyanin, you can see the similar result. Flavonoid, you can see the similar result, and in the case of protein as well. Then in the case of ascorbate and glutathione, the plants are the seeds are seem to be, in the case of UVB prime, they appear to be less stressed. That's why there is low production of ascorbate and glutathione, which are non-enzymatic antioxidants in the primed treated plants. Now we have total amino acids. It is more in heat stress plants. Then total sugar. Sugar is more in the case of UVB primed plants. Phenolics is also more in the UV primed plants. And proline, as you all know, that they are stress indicators, and it is more in the heat stress plants than the UVB primed plants. Then coming to the enzymatic antioxidants, we have superoxide dismutase, catalase, APX, and POD. And in the case of catalase, POD, and APX, here also you can see the plants appear to be less stressed. Thus, that in the case of UVB applied plants, that's why this HS plus UVB, that is the UVB primed plants, have less enzymatic antioxidants. Then I went for the LCMS studies. Here, in the case of, you can clearly see that, as I told before, when the UVB is applied, they have better germination rate. And this better germination rate is because of the GA3, gibberellic acid. And this gibberellic acid is more in the case of UVB primed plants. And using this gibberellic acid, plants are not going to thermal dormancy. They are overcoming that thermal dormancy. And in the case of ABA, you can see more ABA in the case of heat stress plants, which means this ABA is the reason for the thermal dormancy. Then, and the same in the case of salicylic acid and not the same, different. In the case of salicylic acid and the isomeric acid, as they all are you all know that when plant is not in a favorable condition, they tend to go for dormancy. And so the HS, so in the case of salicylic acid and jasmonic acid, when heat stress is alone applied, those plants produce more of these hormones, which causes the dormancy. But in the case of heat stress UVB applied plants, the levels of this jasmonic acid and salicylic acid is less, so they can easily germinate as well. So I'm moving to the conclusion. So the studies that seeds treated with UVB radiation exhibits improved seedling establishment, as evidenced by the increased shoot length, root length, dry weight percentage, and seedling vigor. And also, you can see a better germination rate and seedling establishment as well. Then we need to conduct more studies, what all things are happening in the seeds regarding the UVB priming. Thank you.
Yeah, nice novel. Within time you have finished. Okay, now the session is open for the questions. Yeah.
Just a quick question first. If you go back to your rice seed hormone profiling, what were the numbers next to the graphs? What are those numbers?
This cell range, those kind of things?
Sorry?
This cell range?
Cell range?
No, the thing is like in my laptop, it is clear. I don't know when it came here. It is showing some kind of cell range, those kind of things. You are asking about cell range, right? Or?
No.
This table. UVB, HS, UV, HS plus UVB, this table.
Okay, no, it's okay. We can talk later. I don't. All right, thanks.
The display got messed up. No, it's not student fault.
So you checked GA3 in seeds,
Seeds,
or plant
Seeds.
at seedling level?
Seeds.
In seeds.
Yeah, at the 60th hour.
So-
after imbibition.
So why you checked only GA3? What about other GAs, which are active form of GAs?
I have looked for GA4 and GA7.
So in all that cases, the expression was higher?
No, the thing is like only a slight difference, not that significant differences there. That's why I didn't put here.
Okay.
Yeah.
Yeah, Professor. Yeah, it's a good presentation. I just want to know, I mean, any priming, not only, the mycorrhiza or, whatever, there is an improvement. So what is the exact mechanism?
The thing is like...
Now you said some ABA also.
That is
but,
one thing.
that is also may not be, exactly the...
in the case of seed dormancy, here it is clear that because of gibberellic acid, they are overcoming the dormancy, and because of higher amount of ABA, the heat stress plants are having more dormancy. So that is the one reason. Then another reason is like you can see that changes in the antioxidants and the non-enzymatic antioxidants, etc. So they may help the plants to produce less ROS, so that plants can better well. Then in some papers they are also telling about this priming can trigger some methylation changes and in the promoter level as well as in gene level, so that the expression of certain genes get enhanced because of this priming.
Yeah. So I have sorry, go ahead.
So just a quick question about the relative doses. So you're giving them a dose of UV. I wondered if you knew how that compared to what the seeds would receive in the field. I know they'd be buried, but the UV will still go through the soil.
Yeah, like the thing is, in the case of rice seeds, in the seedlings, it depends upon different regions how UVB is getting affected. Yeah, but in the case of seeds, actually, in the case of rice, we will put in the soil. Then they will not get that much UVB there. I don't know.
Okay, not much, but what is it? Are you giving them 100% more or 50%? How much more than they were receiving in this field? What is your dose? Is it 10,000 times more or just 50 or what? It would be nice to know the comparison.
Actually, I don't have that.
No, it's just for future. I'd like to know. Thank you.
One query. Have you used the dry seed or wet seed for the UVB priming?
Like I will take the seeds, then after surface sterilization, I will go for germination. I will sow in the filter papers. So at that time itself, they started their imbibition phase. Then after 20th hour only, I will give UVB priming at that stage.
Yeah, Mike. Okay. Why you chose UV as a priming agent? Why not directly heat as a priming agent?
UVB, right? Actually, in my lab, this UVB priming studies is already there. In the case of salinity and all those salinity, drought, etc. So, and I look for some papers. They are telling about heat priming versus heat stress. So because of that, I choose whether any UVB have any effect. Then I checked for that. That's why I got the result. That's why I did it.
Yeah, that's why my query was in other way, because UV itself induces some modification in the methylations and all those things.
So it may be confusing sometimes whether it is because of UV or it is because of priming the results you are getting. But it will be interesting to see the results between
like
that.
In next stage of study, I want to study the methylation studies, because any kind of like what kind of changes this UV is doing in the DNA level. So from that, it was we will get that clear picture about this thing. And other thing is like from the phenotype, it is understood clear that the plant is not getting that much harm because of this low dosage of UVB.
I have one question here. So why did you choose 45 degrees? It's very high temperature actually as a heat stress.
In the case of rice, already there is lot of papers based on like they will give 45 hours for the stress, and the rice itself is a heat tolerant plant. So like the thing is like it is stated that 28 degrees is the optimal for the rice, but I go from like from 35 to 50. So for priming studies, there is one kind of formula is that plant should have 50% of growth reduction. In this case of germination reduction, then only that priming will affect. We can't go for higher and we can't go for that lower, because in the case of lower stages, when we apply the priming, we will not get that difference between the treatments.
So you are subjecting plants to extra stress basically to see the effect of priming.
Yeah, the thing is like in some stages, like I used to be in Andhra, so there I can experience the temperature between the 45 to 49. It is there, and in some other places as well.
Okay.
Are you confirm rice is heat tolerant?
Actually, now we are just time limitations, so we can discuss while during lunch or maybe other time. Okay, so now it is a time to close this session. Okay, so it is very good presentations by the faculty and all the students. Thank you.
There are few announcements that Josh will announce now.
Yeah, I'll announce from here. So, hope you all know that you have a YouTube streaming is going on. Maybe that has been shared with every participant. You can, if you want, you can just share it with your colleagues or someone who would like to interested in this. Then at 4:20 to 6:20, we have the poster session evaluation. So, I request the seniors over here, you should support the students. All, please watch the posters which will be there in the block next to this. And thirdly, we have a Wi-Fi password. So if at all nobody has received that, that can be obtained from the registration counter. I hope most of them know about it. And some were asking about certificates. So we are going green. So we have the certificates which can be downloaded from your, you can scan the QR code on your, your identity card. Yeah, then you can download from tomorrow noon onwards. It will be available only from tomorrow noon. So we are a little cautious on that. Okay. Then regarding the duty leave certificate, some have already given the request in the counter. They can have it either today or tomorrow. They can have it. And lastly, today lunch is special with Kerala style. You have to sit and it will be served. It is not buffet style. So other team has already come up there. So we have to rush immediately. Otherwise, it has to be done in a one stretch. It has to go in one stretch. Okay. Thank you. So I think there is winding. Nobody has given thanks for the chairs. Yeah, that will come from that end, I suppose. Yeah, Ria.
So thank you all for the wonderful session. And I, with most humbly, I will thank the chair and the co-chair for very wonderfully conducting the session. Thank you all.
Thank you, Ria.
Yeah.
I'll make an announcement. Ladies and gentlemen, the session will be delayed 10 minutes due to computer technical issues. Don't worry, we'll keep the time. Good morning, good afternoon, everybody. Welcome to the nanoparticle plant interaction session. And let's welcome our first speaker, Dr. Liz Rilot, who will be speaking about controlling implanted gold nanoparticle synthesis and size for catalysis. I'll make an announcement. We have issues with the computer. So after this talk, they have to change the computer. We will have 5 minutes, 5 minutes delay. I apologize.
We're on. Hello, everybody. Can we hear you? Hooray! Thank you. Well, here's my spotty dog story. Using synthetic biology to manipulate the size and shape of gold nanoparticles. Why would you do this? Sustainability of platinum group metals and gold, it's an issue. We use it a lot in emerging technologies like your mobile phone and your laptop. And more of us want mobile phones and laptops. Certainly my kids do. We've got no effective substitutes for some of these elements, so you can't use zinc instead of palladium or platinum instead of palladium sometimes. They're not interchangeable. You have to use that element. So we've got no effective substitutes. There's a high adverse environmental footprint when you mine these. They're very, very carbon footprint heavy. So once they're out, you want to kind of reuse them. And the reserves are dwindling, and I can't quite see there, but the natural deposits are geopolitically concentrated. Is that going to turn? Okay, so here's global distribution. I say global, but the Americans have got a good chunk. Russia's got a lot. I don't think they'll be going to hand it over very readily. South Africa have got a good chunk as well. So platinum supply is not equal. The reserves that we do have, we need to use them responsibly. Reuse, recycle. Okay. So where have all these elements gone? What's happened to these supplies? We have dispersed them willy-nilly across the planet in some very creative ways. For example, catalytic converters. So at the end of a vehicle's life, the catalytic converter is taken off and the platinum group metals are recycled. But by that point, at least half of them have been distributed willy-nilly across the roads on the roadside verges, and they've been lost, which is an excellent way to distribute a metal in a way that's very difficult to get it back in. They're also used in electronics, dentistry, chemical catalysts, so there's a lot of different uses for them. Okay, and here is a picture of a mine tailing site in British Columbia, a palladium site. And if you can see on the image on the right-hand side there, you can see a tailing dam has burst. And this meant that the tailings, which contain toxic metals and very high concentrations, leached into the river and caused a catastrophic environmental incident. Okay, so that's a problem. There's also a huge problem in developing countries where historically mining has happened and companies have now left this. There's no legacy for cleanup. These have just been abandoned and they're causing huge negative impacts. Okay, so mine tailing sites were previously vegetated before we came in and made this mess. Okay, so can we revegetate them? Environmental restoration is great. Tourism, farming, environmentally friendly, it's good for everybody. But it costs money. Can we use phytomining as a way to finance this restoration? Okay, so we're going to look, do plants even take up these metals? Well, they do. Plants take up gold and palladium. And I talk interchangeably between the two because a lot of our studies were done in gold. Gold is very electron dense, so it's very easy for our experimental procedures, while it's very chemically similar to gold. So we used gold, similar to palladium. So we use gold as an exemplar, if you like. They form nanoparticles in the tissues. You can see the little nanoparticles here. And we can carbonize this plant biomass and use it as a chemical catalyst. So palladium on carbon catalysts are worth 5 times the price of pure palladium. So we could potentially leverage financial money to help environmental restoration. Okay. They also have reduced energy inputs and processing costs. And we did some LCA analysis with Tom Goodell at Yale University. So this is our experimental system. Here we've switched back to palladium, but you can see over the time course as we dose the plants with palladium, and this is in flasks. Admittedly, it's not in a field trial with real level metals, but we'll get into that. This is just proof of concept. Does it work? And you can see with time, we get an increase in the diameter and the number of nanoparticles. So it's working. Nanoparticles are being formed. I said that we could use that to make a catalyst, and it's a very simple pyrolysis step. You take your waste, you grow your plant, you stick it in a microwave oven, essentially, under nitrogen, and you make your catalyst. So it's a carbonized material with nanoparticles of your platinum or gold on it. We can then see if we can do assays on this, if we can have activity using this catalyst. And so we made two different pyrolysis materials, one at 300 degrees C and one at 800 degrees C. And we found that the ones that were heated for higher had a larger diameter of the nanoparticle size, which isn't necessarily what you want, but we wanted to find out what was optimum. Okay, so using these two different materials, we had a look to see if they could catalyze Heck reactions. So this is a coupling of iodobenzene with methyl acrylate to make methylcinnamide. And you can see here that the palladium 300, the 300 degree C catalyst, is highly active. At this point, we ditched the 800. The nanoparticles are too big, and we're not interested. So we'll park those and leave them, and we'll focus on the PD 300. Okay. So then we did 3 different reactions. You don't really need to know what the reactions were, but what we were doing was we're going to see if our plant-based catalyst is comparable to something that you might buy from Sigma, for example. Does it work? And it does work. So we can see that these bars are the yellow, green, and pink bars on the end. There's different percentages of conversion from the substrate to the product, comparing our PD300 with palladium on carbon, for example, which is a commercially available, commonly used catalyst. And we're there. But don't forget, this is in a flask that we've dosed with palladium salts. It's not in the real world, but it looks like it could work if we get the levels high enough in the plants. And what's high enough? How much palladium would we need to get in a plant to make a catalyst that's commercially viable? Well, we can see here that it's admittedly a very high amount. It's about 1 millimolar, which is a crazy, crazy number, thinking that we could pull that out of the mining. But you've got to know if something's going to work before you spend too long on it. So, okay, it's quite high, but let's see. Let's see where we get on. Okay, can we use synthetic biology to manipulate or increase the formation of those nanoparticles in the tissues? Do we want to increase the uptake of platinum group metals? And there's a different project I'm not talking about today, looking at the transporters and the solubilization. But can we increase the nanoparticle deposition, and also can we improve the catalytic activity? Okay, the catalytic activity can be improved if you tweak the size of the nanoparticles. So smaller is better. Smaller nanoparticles have a larger surface area, and also they have a higher turnover time. You can reuse the catalyst more times because they don't fall off the catalyst. The larger ones fall off, they're heavier. You can't use them so much. So what we did was we expressed synthetic peptides that bind gold ions in Arabidopsis. Okay, so this is our proposed peptide experiment. This is based on some work done by Tan et al. And Brown et al., where they did some phage display screening to find synthetic peptide sequences that would bind in vitro gold nanoparticles. So they have peptides, a beaker full of gold, and they stir them and they look under the microscope. And this is what they saw. These are not my results, these ones here. So they saw with different nanoparticles, you can see different peptides, you can see different nanoparticle sizes. And it all hinges on the 2 central amino acids that you have in your peptide. Okay, so we've got a pointer. Okay, on the giant, on the side there, you can see you've got 2 tyrosines in the middle. The tyrosines have real binding activity. They pull the ions in, bind them, so you get very big nanoparticles. We're calling them giant nanoparticles. And you go right the other side to the weak, you've got 2 Ws, okay? Ws are tryptophan. They have weak binding, but they have strong pulling, strong reducing activity, okay? And they make much, much smaller nanoparticles. So that's quite interesting. Well, I'm a plant scientist. Let's put this into Arabidopsis and see what it does. So we did a very simple cloning experiment. We've got a 35S. Oh, yeah, we can get rid of them. Sorry, guys. Why? There we go. Go away. The last one. That one. No. Oh, sorry. I'll come here and do this. Okay, so we put it into Arabidopsis with a 35S promoter, so expressing constitutively throughout the plant. Okay. Bye, guys. Thank you. We made some lines. Have I lost my... Thank you. And we checked we had expression. We did a quick PCR, and we do have expression of all the lines in the Arabidopsis. So the next step is, can we visualize these TEMs in the plastid? Did it work? Were the nanoparticles bigger or smaller? Okay. So this is what they look like under the transmission electron microscope. We can see the nanoparticles. They were throughout the cell, but we chose to focus on the density in the plastid, the chloroplasts, okay? So, I now want you to play a little game called Play Your Spots Right. This is based on a very cheesy UK quiz game called Play Your Cards Right, where you have to guess if it's higher or lower, okay? So here are the plant lines expressing the giant peptide, the peptide that should make giant nanoparticles, okay? We had 3 independently transformed lines, and we did the analysis on all 3, and you can see at the top we've got wild type. So if you focus on the TEM spots, see the spots before your eyes, let me know if they're bigger or smaller with the next one, okay? Bigger, smaller? Nah. This is the medium and the small. It's bigger, smaller? I'll go back. Okay, giant. Are you not convinced? This is the importance of statistical analysis, guys, that you need to measure these things, work out the diameters, and then do some stats on it. So if you can't spot the difference, statistics can help you. No, where's my asterisks gone? There are statistically significant differences. And if you look at the graph here on your right-hand side, you can see that there are more smaller nanoparticles in that one with the peptide expressing that should make smaller nanoparticles. And we're seeing giant ones in the giant. So it does work. We're quite excited. That's the cool bit. So what's the next step? Well, let's just go back one. The next step would be to test the pyrolysis of these, and we see that the plant biomass from the small nanoparticles does indeed produce more catalytic activity. So it works. This is great. The giant, it doesn't have such high catalytic activity. So by introducing this peptide into the plant, we can tweak the size of those nanoparticles. We can alter the catalytic activity, and it can make the catalyst worth more because it does more activity and it will last more times. So if we could put that into our final species, that would be something we've learned. Okay, what next? Well, we noticed there's some triangles in our nanoparticles. They're not all spherical. You can see on the screen there's quite a few triangular ones. Now, I'm not a chemist, but the chemists tell me that triangles are especially good for catalysis. They like to pop off the corner. Someone's nodding, it happens. So if we could make triangular peptides, how cool would that be? So we're going to investigate that a bit more. The next steps as well is, I haven't really heard many people talking about this yet, but plant biomass is like the new crude oil. Instead of just taking your crude oil and fractionating for paints, lubricants, etc., you take your plant biomass and you fractionate that, and it has all the same things in it. You have your lubricants and paints from your lignin, through your aromatic compounds, and what's left, your cellulose, if you break that down, you have sugars, you ferment those, you have biofuels. So we could just burn this, like cavemen, and get some heat. Or we could be clever, and we could use the plant biomass to make all these other factors that have got value. Platform chemicals, biofuel. This is cash now, cash and a catalyst at the end of it all, okay? And if you look here, the presence of the palladium, and I also have nickel on this table, so if plants have taken up the palladium, it alters the products that are formed from that plant biomass. The presence of that catalytically active palladium means we're getting off higher levels of some of these compounds that are valuable. So wow, this was an added bonus that we didn't really expect. We kind of thought it might be there, and it is. So that's something else that we can investigate. Not only can we have a catalyst that's got small nanoparticles, so it's really good, we can get some valuable stuff out of the plant matter as well. Wow, we like it. We're really happy. Okay, so this was published recently. I've got a couple of barcode things that you can scan from the room if you wanted, or you can see my talk online later, and you can look at this. So this is work in ES&T that just come out, and a recent review that I put into Science last year. So that's more or less it. The final step would be to look at this in a remediation relevant species. So we're now developing transformation protocols for willow. We know what we've got to put in. It's a tree, and it's going to take us a while. So next talk, willow. Thank you very much for listening.
Thank you, Liz. Questions?
Yes, I do have. Excellent talk, Liz. And I have 2 questions, quick question though.
first is because you are putting this synthetic peptide, which is 8-9 amino acids.
Mhm.
So how they remain stable in the plants, because those will be target for degradation, kind of that. And second is when you have this mine tailings and all those, when you grow these plants that you are expressing these peptides to take up palladium, then what about in the mine tailings there will be other toxic metals as well, and then how your plants will deal with those?
Yeah, no, very good questions as always. Thank you. The peptides, we know, well, we believe that they are expressed stably because we're seeing a phenotype at the end of the day, and we have multiple biological replicates, and we see qPCR that it's expressed. We tried to do LCMS to identify the peptides, and we couldn't find the peptides, but they are very small. So yeah, we're not sure what's happening in that middle stage. We don't know how long they hang around, but they hang around long enough for us to see a difference in the nanoparticle size. So, and we don't know if they're at the center of the nanoparticle, if it's a single peptide which nucleates around, or if it's a cluster.
Yeah, because the mRNA transcript is the target for degradation, because that's very small and become like a RNA mechanism, or that will be activating into that one. So these are the targets for that. So the expression could come, but beyond that, what will happen? And maybe because if they are translated, and then the palladium or gold is bound on both sides of the end of the peptide, then it may stabilize. That might be the...
Yeah, it might do, you're right. Yeah. Okay. So it seems to work, but we don't know how stable they are.
And the second part?
The second part was the heavy metals in the mine tailings. It's a good point as well. There are heavy metals there. And I said, I didn't really talk about the transporters, but we know there are transporters that are specific for gold and palladium that we've characterized, that we're in the process of publishing. So we'll be looking for a synthetic biology approach to localize those in the correct place within the plant and upregulate them, potentially downregulate some other transporters. But we're looking at willow, which has got really high resistance to a lot of different heavy metals anyway, so we might be able to pull that one off without the need for that. But we'll have to see. I think the bottom line is we clearly need to bring in a lot of organic matter into the sites as well to establish the plants.
Thank you.
More questions? No? Okay. Thank you, Liz.
Oh, okay. That online talk is there?
Yes.
Hello everyone, my name is Jay Yang, a postdoctoral research from team of Professor Yu Mingluo and come from Institute of Soil Science Chinese Academy of Sciences. I have been commissioned by Professor Yu Mingluo to show our work. The title of my presentation is Effects of Klein Minerals on Weight Uptake and Transport of Nanoplastics. There are 4 minor parts into this topic. Start from background, micro nanoplastic pollution. Then I will introduce our specific content, Klein Minerals' particular effects on uptake of nanoplastic in weight settling, and weight can uptake regularly shape nanoparticles. Finally, I will show the conclusion and pose the prospect of our research. Firstly, we will come to the background section. Microplastics are commonly defined as plastic particles less than 5 millimeters in size and are widely distributed throughout the terrestrial ecosystem. The shape of microplastic include pellet, foam, fiber, fragment, film. The exact definition of nanoplastic is still controversial and needs to be standardized, and it is now generally accepted that the size range of microplastic is 1 micrometer to 5 millimeter, and the size range of nanoplastic is less than 1 micrometer. Soil is a major sink of microplastic pollution. Microplastic pollution in soil is an emerging environmental issue. They have various sources, such as breakdown of larger plastic, microbeads from personal care products, terrestrial fraction, and synthetic fiber from textiles, and migration by wind, rain, and human activity. Microplastic pollution are widely detected in marine and freshwater environment. As a major recipient added to farmlands in European and North America each year is estimated to be much higher than the estimated annual global burden in ocean water. Those microplastics can protect potential risk on ecological and environmental health. In China and other countries around the world, the use of plastic agricultural film is very common in vegetable cultivation. Our previous study reported for the first time that polystyrene PS nanoplastic 200 nanometers can be taken up by root apex, where PS microplastic 2 millimeter can enter crops through cracks in the laterite root sections. Crops can uptake and transport micro nanoplastic, and we used EU complexity dumped nanoplastic to achieve quantitative and visual analysis of nanoplastic uptake by crops. Studies has indicated that many terrestrial plant species can take up and transport micro and nanoplastic to the above tissues. However, little is known about the effect of clay minerals on the uptake of micro and nanoplastic microplants. In soil crop systems, both crops and clay minerals can reduce the migration of micro nanoplastic and affect the environment behaviors of micro nanoplastics. The hydroxyl groups lead to the difference of the interaction between micro nanoplastic and the different ion oxygens and promotes the different heterogeneous agglomeration of micro nanoplastics. Clay minerals form heterogeneous aggregates with micro nanoplastics can promote photodegradation of micro and nanoplastics. However, little is known about the interaction of mineral particles with micro nanoplastic in soil plant systems. So we pose the question: how to clay mineral particles effect on the uptake of nanoplastic in wet settings, and whether plant can take up irregular shape mineral particles. Okay, now I will introduce our research content. We designed the experiment 1 and nanoplastic co-exposed experiments. This experiment investigated the influence of two common clay minerals, colinate and montomerinolite, on the uptake and transport of PS nanoplastics by wet settling in hydroponic solutions. The concentration of PS was 40 mg per liter, and the concentration of clay minerals was 1 grams per liter. Our previous studies used fluorescence labeling and scanning electron microscope reveal that micro and nanoplastics enter root stainings at laterite root emergency, followed by transport to shoot. The content of EU label PS show a good correlation with EU. The results show that the fluorescence intensity in root apex, laterate roots and primary roots from the PS plus the coordinates and PS plus the Montgomery treatment was lower than that in the PS treatment. Minor decreased the uptake of microplastic in weight from laterate root cracks, especially when nanoplastic and Montgomery coexisted. The larger specific surface area and the stronger cation exchange capacity makes the adsorption capacity of Montgomery stronger than coordinate. In primary roots, when Klein minimals are present, the nanoplastic transport in weight vascular tissue decreased, especially when nanoplastic and Montgomery coexist. Those results indicated that the presence of Klein minimals reduced the uptake of PS nanoplastic by weight roots, particularly when PS nanoplastic and Montgomery coexist. Difference in the uptake of PS nanoplastic by plants may be related to the state of PS nanoplastic in suspension. When PS nanoplastic was exposed with Klein minimals, they can deposit on the surface of Klein minimals. Those aggregates promote the settling of nanoplastic, reducing the uptake by plants. Electronic interaction are crucial for the stability of the suspensions. SCM emergent further reveals the adhesion of PS nanoplastic on the root surface. Plant roots has a negative change due to oxygen containing fraction groups such as hydroxyl and carbon oxyl, and adsorb the external substances through electrostatic interaction and adhesion of exudates. The presence of adhesive substances on the root surface captures and immobilized the hydroaggregates formed by the minors and the nanoplastics. Quantitative analysis by EUPS microplastic revealed that the weight root treated with EUPS nanoplastic could take up 3,950.7 to 4,242.8 milligram per kilogram, and the content of nanoplastic transport to shoot was 70.7 to 78.3 milligram per kilogram. Klein minimals decrease the uptake of nanoplastic in weight, especially in coexisted to Montgomery particulars. Klein minimals has similar size with microplastic, so whether plant uptake irregular shape nanoparticles. The uptake of Klein minimals by plants was traced through fluorescence techniques. The surface of Klein minimals was chemically modified through covalent bonding in this study. This means equated the high stability of labeled particles. We designed the experiment too. Klein minimals expose experiments. In this experiment, we used the fluorescence labeled Klein minimals and Paris Klein minimals, and we adjust the exposure time according to different army. The fluorescence signals of two minimal particles was located on the cell walls of the critical tissues in root apex. With increasing exposure time, high fluorescence signals exhibit a clear creek enter mode into the vascular system after 96 hours, particulars at 168 hours. The fluorescence signals of montomineralized particulars was located primary root on the cell walls of the root cortical tissues. The SCM emergence indicated the locations of the suspect Klein-Minimus particulars in wheat roots. We further use SCM to search in the Klein-Minimus particulars in stems and leaves. Those particulars exhibit small surface compared to initial particulars in the root. Furthermore, we analyzed the Klein-Minimus in xenon shape by STM. Many Klein-Minimus particulars had inorganic nutrients and organic matter attached to their surface, such as phosphorus, potassium, and iron. STM showed that those particulars had crystal structures. Line scanner profiles of the carbon elements in some Klein-Minimus particulars in xenon shape revealed carbon content on the particular surface. Additional AFM-AR was applied to study the fractional group of the surface contents on those particulars. Take montomineralized treatment as example. The infrared spectra of pristine montomineralized showed a strong and shape single peak at 1061 was attributed to silicon and silicon oxygen stretching vibration. We found the suspicious particulars in xenon shape has this peak in 1068. Further confirmed that the particle is montomineralized. I will conclude this work. Klein-Minimus can reduce the uptake of nanoplastic in weight by forming heterogeneous aggregates with nanoplastic or competing for uptake pathway in the new lateral root crux. Montomineralized due to its larger size and specific surface area shows a more pronounced effects in limiting plant uptake of nanoplastic compared with covalent living and advanced microscope revealed that Klein-Minimus and roots status at lateral root emergency seeds, followed by transport to shoots. In future, I think we need to explore the maximal size range of particular uptake by different crop roots, and also the core transport process and mechanism of Klein-Minimus and micro nanoplastics. We also need to investigate the positive and negative impacts of mineral particulars on crop growth, nutrient cycling, and pollutant transport, and also to explore the interaction of crops plant with other colloidal particulars in soil, including colloidal phosphorus and heavy metals. This work was supported by the Ministry of Science Technology of the People's Republic of China, National Natural Science Foundation of China, and the Chinese Academy of Science. That is all my presentation. Thank you for your attention.
Okay, now we move to the next presentation. Dr. Rahul Kumar from the University of Hyderabad Telangana, we will speak about Creo Millet Nano DAPA for enhanced plant growth, sustainable agriculture.
Good afternoon. First of all, I would like to thank the organizers, Professor Josh, for giving me this opportunity to present a part of our work here in this theme, where we have generated some nano fertilizer, especially phosphate fertilizer. And in this talk, actually, I am going to take you through some of the published results and as well as some unpublished results. So the talk will be like we have generated the material, then its characterization for its properties, and then in the last I will be discussing its effect on plant growth and development. So why phosphate? The slide itself shows you that if we don't provide phosphate to a cell, any living cell, then whatever is there on the left side here, it will happen that the plant will not grow well. Especially crops plant, because then we lose yield potential. Okay, if you look at the green revolution, which started in 1960s, what is the progress in terms of fertilizers consumption? I'm taking three big agronomic economies: China, India, and US, and you will see that there is a consistent increase in the usage of chemical fertilizers altogether, NPK and other micronutrients. Now, what has happened in last 60 years, because phosphate fertilizer, so phosphate is mined from phosphate rocks, which are naturally formed, have taken a lot, millions of years in the formation, but in last 60, 70 years, we have exploited them for mining purpose a lot. And right now, we are in the era of peak phosphate production, which we will peak or expected to peak in 2033. And then there will be a decline, and this decline will ultimately lead to exhaustion of this resource from Earth. The rough estimate, there are different estimates, but I'm taking the longest estimate, which is 300 to 400 years. Just imagine after 400 years, the nature of our agriculture, it is going to see a big shift. Okay, now what can be done in this regard? So, because these chemical fertilizers, they are not highly nutrient, their efficiency is not quite great, because whatever we apply to the farms, only 15 to 20% is taken up as a phosphate fertilizer by the plants, and remaining P is actually fixed in through the soil because of its high reactivity into P metal complexes. It is also immobilized or taken up by microorganisms to support their growth, and then it is also absorbed at soil particles. So basically almost 80% of the phosphate which we apply to the plant as fertilizer is not being utilized by the plant, and it has its own environmental consequences, like with runoff water, the remaining phosphate goes to the water streams and then cause eutrophication.
Now, the question here at hand is, can we improve the nutrient use efficiency of any fertilizer? In this case, in my case, we are working on phosphate fertilizers. So then in 2020, we started, along with my friends from material sciences, we just, are trying to ask a question, can we really improve the nutrient use efficiency by reducing the size of the currently available phosphate fertilizers. So we took the granular form of this phosphate fertilizer, and then first thing was that we simply milled them. The problem with milling was that there was very rapid re-agglomeration of these particles, so milling was not as efficient as we were thinking. Then we proceed to cryomilling of that, which is milling but under liquid nitrogen conditions or highly lower temperature. In this case, what we realized, so we milled for 1 hour, 2 hour and 3 hour, and then we characterize the material here in FVSEM. So you will see 1 hour mill, 2 hour mill and 3 hour mill sample. When we characterize them, so we found that the 3 hour mill samples, they showed the best size reduction in case of these particles, which we by DLS we estimated average size is 334 nanometer, whereas 2 hour mill and 3 hour mill were more than 700 and 900 nanometers size. Then we checked its solubility of the material. So because the milling might not have interfered with the basic properties of this fertilizer. So we found that nano, so I will call commercial DAP as CDAP from now on, and nano DAP as NDAP. So what we found that NDAP was having or showing better solubility in water than CDAP, which was correlated or well supported by the FTIR spectrum data, where we found that these two peaks, carbonate peak, which, so if it is steep, then it basically shows that its carbonate peak will interfere with the solubility of the product in the water. So the CDAP, basically this line, it has very steep peak of carbonate indicating little less solubility in water, whereas the nano formulations 1 hour, 2 hour, 3 hour, they were having smaller carbonate peaks, indicating or which was correlating well with the higher solubility of these nano products. Additionally, the phosphate peak or HPO4 peak was also found to be better than this one, basically, in the nano products than the CDAP, indicating that somehow this cryomilling has resulted in more number of phosphate coming to the surface of the product, which may have a better effect on plant growth. That was our idea. So we also checked the PI release from these formulations and found that the PI release from nano formulation was slower than the CDAP, indicating a slower release of the product. And also we checked the stability of this product on room temperature by dissolving it in water and then checking it, or by putting it in the lab open space and checking the moisture content, which indicated that the product is stable because it is accumulating less moisture than its granular form or commercial DAP form. Now, once it looked like a better product in comparison to CDAP. Then I just forgot one thing to tell. So overall, if you look at this, so by 3 hour cryomilling, we were able to reduce the size of the product by 378 from 5 millimeters, so approximately 5,000 times size was reduced, and at the same time it increased actually the product specific surface area increased by 14,000, making, and then after that better solubility and this phosphate peak, which is we predict has come to the surface, makes it a better product, but is what yet to be tested on plants. So we took one monocot and one dicot plant. I am not actually sharing the complete story. We used soil mixing method, foliar spray method, and then we also put it in the water and then sprayed on the plant or provided from the bottom. So we use different methods. Today I am going to show only the soil mixing method results. So basically when we checked their performance on plant growth, so we took a 50% reduction of the field recommended dose, and on the left side 0P 100% KH2PO4, which is our the salt which we use in tissue culture, plant tissue culture, then LR grade Dept, which is 100% pure phosphate source, and here it is 50% field recommended dose supplementation of commercial Dept, C-Dept, 100% C-Dept, and here 1 hour, 2 hour, 3 hour mill sample. And you will see that the 3 hour mill sample, even first and second hour were also giving good result, but we saw the best plant growth promotion in case of 3 hour milled sample. Then we took this sample 3 hour and then further checked different doses, 10%, 25%, 50% and 100% of C-Dept, and compared with the N-Dept 10%, 25%, 50% and 100% C-Dept at seedling stage. And what we found that the N-Dept grown plants at each concentration were showing better shoot length, they showed better shoot fresh weight, and they also showed better quantum yield, Fe by Fm, then C-Dept grown plants, and not to our surprise, because we were expecting that from where this better growth will come actually. So when we checked the PI content, soluble PI content in plants in root and shoot, we found that the PI levels, total soluble PI levels were actually enhanced in root and shoot of N-Dept plant at each concentration of some supplementation than their respective controls, right? So then if photosynthetic efficiency is better, then the carbon exchange or carbon dioxide exchange rate will also be better. Then we also estimated the number of stomata in these plants and found that there was a consistent increase in the number of stomata in matured, youngest, fully evolved leaf top leaf actually at each concentration, 25%, 50% and 100%. Right now, so this experiment actually, I don't know, we have conducted in controlled condition maybe more than 20-30 times, and every time there is the repetition of the results, but then there was time that we should now test its potential on yield. So what we did that, so phosphorus is actually present in the top layers of soil, the maximum concentration. So what we did that we went to digging site, the field, removed first 2 feet layer soil, which is basically enriched in phosphate source because of the supplementation of these chemical phosphate fertilizers over a long period of time. And then we took the soil from 2 feet deep dig, soil, which actually we tested the phosphate concentration there, it was really low. And then we used that soil to conduct the pot based experiments in greenhouse condition with 25%, 50%, 100% C-Dept and Dept, and 100% KH2PO4 and 0P was always our positive and negative control. Now I'm not showing the growth parameter data, but I'm directly taking you to the yield data. Top layer, top panel 0P, 25% C-Dept, 50% C-Dept and 100% C-Dept. Even after 35 days of the growth, they were 100% C-Dept. You can see a few fruits here. 50% C-Dept, some flowers, here you can see some fruits, but if you look at the panel, bottom panel, then you will see that 25% N-Dept, 50% N-Dept and 100% C-Dept. N-Dept, they are actually showing more fruits than any of the concentration of C-Dept which we used in the study. Even 25% N-Dept actually grown plants are showing better fruit number than the 100% C-Dept grown plant. So this is season 1 data, this is season 2 data, summer season and winter season. Again, we found that consistent improvement of fruit number and yield basically when we were supplying this N-Dept to the plants in comparison to their corresponding Dept controls. Okay, so then we calculated the average number of fruits per pot and then average fresh weight of fruits per pot, and finally we calculated the average yield, and we found that 25%, 50% and 100% all were giving us better yield. And if you compare 50% of Dept is actually outsmarting the 100% of Dept is outsmarting 100% of Dept in terms of yield. So we actually, by reducing the 50% of the dosage, we were able to have similar number of fruits or same yield coming out from Dept grown plants. Now we are trying to understand what is happening in those plants. One submission, we don't know actually whether it is a effect of higher P or there is some property coming from nano particles which is inducing the plant growth. So we are trying to devise some assays where we can understand this phenomenon. But my lab also works in phosphate starvation and molecular part of this phosphorus starvation and signaling. And in our data consistently we find a ribosomal S6 kinase gene which is activated or induced upon low phosphate availability. So this, then we thought, let's check its expression. So that is basically some RNA-seq data, and this gene actually is induced or upregulated under low phosphate condition. So then we check this gene, and this is, by the way, this gene belongs to TOR signaling pathway, which control plant growth and development. So, and we checked the expression of this gene at 50% NDAP and 50% CDAP, and again this 50% CDAP, we found this gene to be induced, indicating, so it's basically a negative correlation, that the better growth in NDAP is most likely coming from its better PI availability than what is available in case of 50% CDAP. And luckily we have antibody for this gene, so we checked the protein level also, and you will see that the qPCR data is on this completely is going on with the lower accumulation of this protein also at the same concentration. Okay, I told you two crops, monocot, dicot. So we are also testing the same formulation on using rice, where actually we for the first time we are growing rice. So we struggled a little bit initially how to grow it, but again you can see that it has the 50% CDAP, NDAP and 100% NDAP are much better, or even 25% NDAP grown rice seedlings are much better than their respective controls in that. These plants also showed better photosynthetic efficiency, and at least at 50% NDAP and 100% NDAP supplemented formulations, they took lower number of days for panicle emergence than their controls. And when we check the average number of panicles per plant or grain per panicle, these yield parameters, we could only see some changes coming at 100% concentration and not 50% concentration the way it was working in case of tomato. There can be again two reasons. Once, we are still learning how to grow plants in greenhouse with us, and second thing is that rice needs more phosphorus, and actually its seed has more phytate level also. I don't know, there may be something which is endogenous to rice and then interfering with the performance of this material. We have to ascertain that. So coming to the conclusion, in this study we actually used cryomilling approach to reduce the size of commercial DAP here, and when we used it to support as a supplementation, we found that the homogeneous availability of PI nano formulation close to roots making the plants grow better and more heat, whereas in case of these granular plants which we take 1 or 2 per pot, so the plants which are close to or roots which are close to that particular granule, they will grow like the NDAP grown plants, but the plants which are away from those granules will sense phosphate deficiency, and that's where we consistently see very uniform growth, seedling growth in case of nano DAP, whereas in case of NDAP, whereas in case of CDAP, some plants are in the same pot, some plants are smaller, some plants are medium height, and some plants are taller, so there is a big range of phenotypes from there. So yeah, so basically our product is actually leading to enhanced PI uptake, enhanced photosynthetic performance, plausibly activation of flower signaling pathway, which leads to better growth. And
Excuse me, your
just
time
finishing,
is up.
I'm just finishing. Yes, acknowledgement to all the authors and contributors and funding. Thank you very much. I'm done.
Time for one quick question.
Yes, I have a question. Over here. Sir, you told me that during, first of all, wonderful talk. You are supplementing the phosphate exogenously. Yeah. So is it going to imbalance of other nutrients over there? I mean, micronutrients. Have you checked about other micronutrients which is going to be taken by the plants? I guess there must be some imbalance. I mean, more of a phosphate rather than
we are
other,
not putting more phosphate. We are putting the same amount of phosphate which we are putting in.
Okay, so have you checked in the plant which has been taken and the nutrition level of other micronutrients?
So PI we have checked. Other nutrients we have not, but since we get better growth, seedling growth, this, and we don't know what is the reason, we have actually now this in plan. What we checked is nitrogen, which is for certain NDAP plants don't show more nitrogen or fixed nitrogen than the CDAP grown plants. But other micronutrients we are yet to check them.
Okay, thank you.
Yeah, nice talk, Rahul. I want to know that since you are talking about phosphorus transport processes, so you got also in the when you are using NDAP, then you are getting higher amount of phosphorus in the plant. So whether they are using the same pathway, how phosphate transporters?
So we checked the expression of these phosphate transporters, and we did not found any change between CDAP and NDAP, at least in terms of their gene expression or transcript level. But it must be, because it is the same thing. I think that so material after cryomilling remains its basic properties. It doesn't change much as a material. But since the more phosphorus peaks are available or exposed to the surface with bigger surface area, that means it is just, plants are simply taking it more. Now there can also be this release part of it, that it is slow release. Maybe that can also be influencing plant growth, but we don't know how.
So they are entering in nano form only, or they are
again we don't know, because phosphorus is immediately, so we thought to tag it with some fluorescent dye, but then we had to put it back in water, and then before it moves to plant it will disassociate. So we are frankly, we don't have any solution to check whether it is PI uptake through transporters or through some nano mechanism. Yeah, can't say.
Can I ask?
Yeah.
Yeah, this is Padmasri.
Madam.
Rahul, very good presentation. Have you tried growing plants in the same soil where phosphate, your NDAP or whatever it is added?
Same soil means what?
One life cycle of the plant, second life cycle of the plant, like that.
No, we don't. We use that soil once, because then phosphate level, we have already put phosphate there, so we can not use it as phosphate deficient. We have not used the same soil in multiple cycles.
Okay, if anybody is interested, you can continue the discussion during the coffee break. We are running late. So please, Dr. Kumar, you can talk to your colleague during the coffee break. Now we have Sudhir Sharma from the University of Massachusetts, who will talk about nanosulfur, a novel nanomaterial for heavy metal mitigation in agriculture for food safety.
it was just 19 minutes.
Sorry, I have to interrupt you.
Is there any other one? So Sudhir is there?
Oh, I thought you changed the laptop.
Yes, we changed it.
So, and this one is working with the.
Yes, so this is Noble's laptop, and we have that one. Which one is yours, Toshiba?
Yeah.
And yeah. Just check it. If it was something. Look. Hopefully it's not the center here.
Yeah, hopefully.
You just check. It is coming intact as you want it, right?
I have to check real quick.
Yeah. Check it. I think it is.
Yeah,
Number.
yeah,
Yes.
Hello. Good afternoon, everyone. My name is Sudhir Sharma. Without taking too much time, I'm going to start with my presentation. So, I will be talking today about nanosulfur as a novel nanomaterial for heavy metal mitigation in agriculture for food safety purposes. So, since we are short of time, I'm going to skip through these initial introduction slides a bit quickly. But as we know, there are several threats for agriculture. As we know, global population is increasing. One thing of notice is that during pandemic, we noted that there was a sudden rise of global hunger. So I'm just showing this to tell that global food security is not as secure as we think it is. Global hunger can increase very rapidly based on many geopolitical issues. So that is something as scientists we always have to keep in mind, and we have to work towards creating buffer so that we can increase our food security and strengthen the system to ensure that. Continuing with threats to global food security, this trend is quite persistent, that many of our major agricultural crops are showing reduction in the annual increase of their production. So this is just to say that we need to increase the production of our major food crop to ensure global food security. And there are certain other factors which sort of compounds these threat levels to global food security. One such important threat is heavy metal contamination in environment. Just a disclaimer, there is no IUPAC definition of heavy metal, but for our purposes, we can say that any metals with high atomic mass, which are toxic at relatively low concentrations, can be considered heavy metals. These two small maps, they're just showing the prevalence of arsenic and cadmium in the topsoil in US. I'm sure there are many other maps present for the whole world, which shows that these heavy metals, they stay in the topsoil and they're quite prevalent. And that's why when humans are exposed to such metals, there are several diseases that are known that happen because of these heavy metals. These are just some examples of disorders and diseases caused by heavy metals. Cancer is among the prominent one. Apart from humans, these heavy metals are also toxic to plants and usually create these kind of issues in plants like ROS stress, lipid peroxidation, decrease in photosynthetic efficiency, overall reduction in plant biomass as well. The problem occurs when these heavy metals accumulate to such a level that it enters our food chain. As it was mentioned earlier in one of the talk by Professor Dhankar, that in recent years there were these two congressional reports which established that many of these major baby food product companies had their food contaminated with dangerous levels of arsenic, cadmium, and other heavy metals. So this just goes to show that heavy metals entering our food chain can create a big trouble, and can cause special worries for vulnerable populations like elderly or children, or especially in this case infant babies. So this is a problem in plants. So what could be a potential solution? Sulfur for the rescue. Now, sulfur in general is quite useful for humanity. It has so many uses. I've listed few here. You can read. For from volcanicization of rubber to use as a pesticide, sulfur is very useful for humankind. More importantly, it is a major macronutrient, and after NP and K, sulfur is a rate-limiting nutrient for many of the plants to achieve their potential growth. But for the case of heavy metal mitigation or heavy metal toxicity mitigation in plants, sulfur is essential because of this one particular biomolecule, which is glutathione. I'm not going to discuss it in detail, but glutathione and its oligomers, phytochelitins, are basically responsible for binding to the xenobiotic heavy metals in plant cytoplasm, and then they take it to the vacuole and sequester it there so that the cytoplasm is safe from the toxicity of these heavy metals. So this slide just tells you how important sulfur can be in mitigating heavy metal stress. When sulfur is that useful, it sort of makes sense to try out nanosulfur, because a normal application of sulfur in soil is in the form of gypsum or on some other form of salts, which are readily available, but unfortunately there is a problem of leaching with such applications. So nanosulfur, on the other hand, could be a potential source of sulfur in field. And my study in my PhD program was to evaluate the potential of nanosulfur to mitigate different heavy metal stress in different crops. Just briefly, nanoparticles are basically anything which are in the realms of 1 to 100 nanometer. I'm sure this has been talked before, but I just wanted to make sure that we know what kind of particles we are talking about. And nanosulfur in particular has been reported to mitigate stresses like mercury stress in Brassica napus, as in this study, and it has been reported to be a very effective amendment to deal with diseases in tomatoes, as mentioned in this study. With this brief introduction, let me just get to what I'm going to talk about as far as the experiments and results are concerned today. So very briefly, I am going to tell you a little bit of story about nanosulfur during my PhD, what kind of plants I worked with, what kind of heavy metals I worked with, and specifically I'm going to talk about these three plant systems: rice, soybean, and wheat, which were examined for evaluation of nanosulfur to deal with the toxicity of arsenic in rice, silver nanoparticle in soybean, and cadmium toxicity in wheat. Let's go to our first experiment. This was to check if nanosulfur amendment in soil can reduce the toxicity of arsenic in rice. So the first experiment was done for a shorter period of time, 35 days, only with seedlings. We had these 6 treatments: no treatment control, nanosulfur, which I'm going to say, or which I'm going to show as NS from here onwards, arsenite at 40 ppm, arsenite plus nanosulfur, arsenate at 60 ppm, and arsenate plus nanosulfur. So this is going to be the general way I'm going to explain my experimental setup from now onwards. I will be showing my treatments here and the general soil properties here, and then we're going to look at the results. So with this general experimental setup clarified, let's see what nanosulfur did to the plants that were treated with arsenite at the same time. So this is how the plants look like, and this is how the results look like. So I want to take your attention here first. As you can see, when there was no arsenic added, nanosulfur was still useful to the plant as it was increasing the biomass. You can see here root biomass, shoot biomass, and shoot height as well. But more importantly, when arsenic was added in the soil, the plants, they grew really small. They had a big yield penalty, biomass penalty. But as soon as we added nanosulfur in the soil, we saw that the plant biomass shot up back to the control levels. So this was clearly showing that the toxicity was somehow elevated because plants were doing much better, almost as good as control plants. Similar trend was observed in case of arsenate, which is another form of arsenic that we tested in this study. Again, addition of nanosulfur reduced the toxicity symptoms in the plants, and the plants were doing better compared to arsenate treatment alone. When we look at the accumulation of arsenic, which is another important thing that you check in such studies, you can see that there was a significant reduction with addition of nanosulfur compared to arsenite treatments alone. That just goes to say that the plants that were treated with nanosulfur were accumulating less arsenic compared to arsenite treated plants alone. When we repeated this experiment till maturity, because we are interested in how much arsenic is being accumulated in seeds, that is the main goal here. We want to reduce the accumulation of arsenic in seeds. We saw, so this is basically the general design. In this experiment, we also added bulk sulfur, which is another form of sulfur, elemental sulfur, to compare whether it was because of the nano size of the sulfur, the results that we were getting in the previous study, or was it just because of the addition of sulfur that arsenic was reduced in the soil and was not being uptaken. So that's why we added bulk sulfur as a control of sulfur treatment in this study. And we repeated this study till maturity to see the accumulation of arsenic in these plants. When we look at the results, the fertilizer effect that we saw in the shorter study was somehow lost. So we didn't see any significant improvement in the plant biomass and seed yield with addition of sulfur alone. But thankfully, with addition of sulfur in presence of arsenite, we were still getting the beneficial effect of nanosulfur. The plants were doing much better compared to arsenite alone treatment, so the toxicity was being alleviated. We didn't see the similar effect in arsenate as we saw previously, but arsenite was still pretty clear. When we look at the accumulation of arsenic in different tissues of the plants, you can see there was a significant reduction in presence of nanosulfur in the plants, in the root, in shoot, and more importantly, there was more than 50% reduction of arsenic in the seeds or the grains of rice plants, which we take as a win because our soil amendment with nanoparticle was reducing significantly the amount of arsenic in seeds, whereas bulk sulfur was not doing so. So it was, you can conclude that it was more because of nanosulfur and the size of the small particles that we were getting this effect. So the key finding from this study, again, I'm not going into much detail, I'm just showing the very basic, very important results of this experiment. We have published this in Environmental Science and Technology. If you want to see what all other experiments and assays we did in this particular study, you can refer to our paper here. In the second study, we wanted to see, will the addition of nanosulfur have any beneficial effect on silver nanoparticle toxicity? You will probably hear more about silver nanoparticle toxicity in upcoming talks. But in short, silver nanoparticles are excellent antimicrobial agents. So they are very effective to kill microbes. And they are used in many industrial products and for many different processes. But the problem arises when these particles end up in field and they start accumulating to such levels that they can affect growth of plants. Specifically, these are highly toxic to legumes because legumes have nodules in them, and nodules are basically symbiotic relationship between bacteria and roots, and these are antimicrobial agent by nature. So they kill those bacteria, and you can see in our study, as I will show, that the plants, they suffer a lot with silver nanoparticle present in the soil. So what we wanted to see was, will addition of nanosulfur reduce the toxicity of silver nanoparticle as well? So the experimental setup was something like this. We used two concentration of silver nanoparticles, 32 and 64 milligram per kilogram. And we also used two concentration of sulfur and two types of sulfur, bulk sulfur and nanosulfur, at two concentrations of 200 ppm and 400 ppm. So with this list of 11 treatments, that's why we had to keep our replicates slightly less. What we saw was quite remarkable. So this is how the plants looked like. You can see here at 32 and 64 ppm of silver nanoparticle, the plants were doing much poorer than control plants. But as soon as we amended the soil with these different sulfur treatments, that toxicity was very much alleviated. But the important results that we saw after this was that at 400 ppm of sulfur, the toxicity was alleviated with nanosulfur only, but not with bulk sulfur, as I will show you in these graphs. As you can see, in general, the toxicity of silver nanoparticle was there at 32 and 64 ppm both. As soon as we added sulfur at 200 ppm of concentration, the toxicity was alleviated, the plants were doing much better, even better than control. But when we increased the concentration to 400 ppm, we saw that only nanosulfur treated plants were doing better compared to bulk sulfur treated plants, which was an interesting result. We saw similar results in other parameters as well. In this case, I wanted to show how the plants were doing. As you can see here, with the addition of silver nanoparticles, the roots were visibly stressed out. There was a lot of necrosis set in, and nodules were not growing as well as control plants also. So that's how we saw that nanoparticle application was actually improving not only photosynthetic efficiency of the plants, but it was also improving the nodule growth. Moreover, when you look at the seed yield, we saw the same trend up to 200 ppm. Both bulk and nanosulfur was useful, but as soon as you increase the concentration to 400 ppm, only nanosulfur was useful and not bulk sulfur. So we can, similarly, we saw that the accumulation of silver was also reduced because of the addition of this sulfur treatment. And we can conclude here that silver nanoparticles were acutely toxic to soybean. Addition of nanosulfur and bulk sulfur helped with that toxicity. But at higher concentration, only bulk sulfur was useful with alleviation of this toxicity. So with that, I would like to conclude my talk. If you have any questions, go ahead. Thank you.
Yeah, we can have one or two quick questions.
Yeah, I just want to ask you about what is this, the mechanism, why, the nanosulfur, how it is, plants are getting up.
Yeah, so based on the studies I have done, I cannot give you a correct or accurate answer because we haven't basically done the right studies to figure the mechanism out. But we have proposed some hypotheses. We know that addition of nanosulfur doesn't release sulfur as quickly as bulk sulfur in the soil. Bulk sulfur we have shown in our paper that it releases sulfur much more quickly, and that may lead to two things. First, it can reduce the pH much more faster than nanosulfur, and secondly, it can also increase the leaching of sulfate, which doesn't allow enough sulfur to be available for the plants to take up. Secondly, in reducing conditions, especially in case of arsenic in rice, there can be formation of arsenic sulfide. So if your nanoparticles, because of their higher surface area, if they can make more arsenic sulfide in the soil, they can stop arsenic from being taken up by the plants and transported to the upper shoot biomass and cause toxicity.
So will be any other heavy metals also it can restrict?
So we tested on cadmium as well, but we got sort of opposite result. So I cannot give a blanket statement that it will help with other heavy metals as well, because it's something that has to be tested without giving any such statement.
Yeah, thank you. Good work. Excellent.
Any other question? Yeah, please.
Yes.
Hi, thanks. Did you have sulfur 200 and 400 only controls? I didn't see those on the graph.
So yeah, we did have them.
Okay.
We didn't put them in this study.
That's fine.
But so what we noticed
Okay,
was
yeah, you had them. They didn't give anything.
No. So we noticed that bulk sulfur at 400 ppm in itself can cause stress in plants.
Okay, I guess that's my next sub question. How can you be sure you're comparing apples and pears, bulk and nano? It's really hard. I'm not sure it's even possible to make that comparison to know what concentration of bulk is comparable to nano.
Yeah, that is true. So we did initially want to figure out how much sulfate is being released by bulk and nano at the same time. But like for the sake of comparing these two treatments, because elemental sulfur is a normal form of fertilizer that farmers do use sometimes, especially when they have to reduce the pH of the soil, or when they have to supply, when they just have to foliarly apply at the time, like if they need sulfur quickly.
Yeah.
So elemental sulfur is a known fertilizer, so we just, for the sake of simplicity, we wanted to...
It might be better to say that you're comparing it to an amount of sulfur that is added by farmers to agricultural soils, rather than say bulk sulfur.
Yeah, so that's how we decided what concentration to go with initially. So,
okay, thank you.
Yes.
What do you think application of more beneficial quality or the yield wise?
So sulfur, I didn't mention it much here, but like certainly there are some crops which are more responsive to addition of sulfur. So I don't think nanosulfur in itself will be extra beneficial to certain types of crops. I think what matters is how nanosulfur is releasing sulfur in the soil. So one problem that it is solving is it is not allowing quick release of sulfate in the soil. So it can provide sulfur slowly over a longer period of time. So that might correlate with more sulfur requirement by certain crops, because some crops might need sulfur at a later stage of their development compared to other crops. So that way it can be useful, but again, it would be conjunction, or it would be something that I'm guessing right now. It has to be tested on different crops before we can say for sure.
Yeah. More field trials are needed.
Yes.
Yeah, I think other questions we can take later, or you can directly discuss with the speaker during the tea time. Can we announce the tea time now? Tea? Yeah, so then we will go to have a cup of tea outside, and then we'll be back in another 15 minutes. Let's say 3:25. 3:15 will be 3:20 then. 3:20, right.
You.
I think we can congratulate all the speakers of this session.
And also thanks to the chairs doing the excellent job. Thank you very much. But the second will continue as well. Yeah.
Thank you.
Good afternoon, everyone. Thank you for the opportunity to present our research on this conference. And as Liz already opened the topic, we can use the biomass of the plants for different aims, to different objectives. Now, what we were trying to do, and that will also connect with the Liz presentation if you were lucky enough to hear it, is we wanted to reimagine, to reutilize plant waste. Why waste anything when we can use it to synthesize nanofertilizers? Now, what we were using to synthesize nanofertilizers was actually cannabis sativa, better known as hemp. Now, why we've chosen that particular plant? Well, because hemp itself, as you can read behind me, is an excellent source of reducing and capping compounds. So many different classes of the chemical compounds can actually serve as reductive and as capping compounds. Some of them are being alkaloids, polyphenols, carbonyl compounds, organic acid, and so on. Now, why use the plant waste? Well, because we are killing two birds with one stone. We can essentially eliminate the waste, and we can get nanofertilizers very cheaply. So, I don't know why this why that happened. Okay. It doesn't work. It was working 2 minutes ago. It doesn't work anymore.
It's stuck.
It's blocked completely.
Just restart.
Sorry for this.
This is stuck. Yeah, close that. Remove the pen, laser. Remove the laser. Just remove it, and then click close that one. Just close it here.
It is not coming.
It is coming. It was there. Here it was. Not going.
It's not going.
Here.
We can open the PDF. We can open the PDF. Just open the PDF.
Okay, just escape.
Yeah. Okay.
Now you go in there. Now yes. Okay. And restart.
Okay. Is it coming? Okay.
Yes. Okay. So the synthesis part is actually quite simple. What we did is we collected what we were considering plant waste, which are essentially stems and leaves of cannabis sativa. We dried them, we performed ethanol extraction, added the salt that we need ion from, and then essentially adjusted the pH if that is necessary, microwave it, and dried the nanoparticles. All this part up to here lasts for 5 or 6 hours, so it is essentially very quick. The only time-consuming part is drying of the nanoparticle, which usually takes either 24 or 48 hours. We prefer to do it for 2 days. So, where the story becomes complicated is when you are actually trying to adjust your conditions. And as Liz already told her in her presentation about in-plant synthesis, in vivo synthesis, that is the part of the whole process that is probably the most complicated one. Why? Because the characteristics of the nanoparticles depend on the several parameters. First thing that you need to adjust and you need to know for your nanoparticles is the pH in which you can synthesize the nanoparticles. And you can see that it was different for the micronutrient nanoparticles that we synthesized. Now, the other thing that will influence the properties of your nanoparticles is the type of salt that you're using for the synthesis. And this is what was very interesting for us. The third The third point on this slide is that also the subspecies or the cultivar of the plant will influence the properties of your nanoparticles. What you can see here, these two figures, represent iron nanoparticles obtained from the subspecies. Okay, I need to step back. Cannabis sativa actually exists as two subspecies. It exists as subspecies sativa and it exists as subspecies indica. Differences between these two subspecies is actually the content of the THC. Subspecies indica has more than 0.3%, while subspecies sativa has less. Now, you can see the TEM images of the iron nanoparticles synthesized from two subspecies. Although they are all spherical because they are relatively small, and this is the reason why we are microwaving. Because microwaving will essentially give you nanoparticles that are less than 30 nanometers in our experience. So, sorry. Okay. So, this is what I was saying. This S is from subspecies, for subspecies sativa, I is for subspecies indica. As you can see, hydrodynamic diameters at the potential, which is essentially the charge of your nanoparticle, as well as the dry size, or the size that you will see on the TM images, are all different depending on the subspecies of the same plant that you are using. So, as you can see, for both, for iron nanoparticles, and it is the same for manganese, zinc, and copper, it's just that we have too many data to present for this such short period of time. It's always the same. Indica will always give you the smaller size of the nanoparticles and more charged nanoparticles than sativa. But there is another thing. You remember that I told you that the salt will also influence the properties of your nanoparticles. Now, this S stands for sulfate. So we use two salts for synthesis of the manganese nanoparticles. We use sulfate and we use nitrate. And as you can see, with nitrate, we are getting less stable nanoparticles because they are less charged. The more charged they are, the more stable they are. And we are getting the nanoparticles, so you can see comparisons, subspecies sativa, subspecies indica, so from nitrate and from sulfate, that you are getting more charged nanoparticles from subspecies sativa and that you are getting more charge from sulfate. I'm seeing something here again. Do I close the rooms or something? Okay, I just closed it. I don't know if I closed also the video transmission. It's blocked again. Let me go out. It's blocked again.
It's locked again. No, what is it?
It's blocked again. I cannot go to the other slide.
So, I'm not sure what's happening. Okay, right?
Okay,
Isn't
perfect.
It?
Yes.
Yeah.
Yes.
So when that comes right, you have to wait for 2 seconds and again start.
Okay.
Because you are presenting on Zoom.
Okay. Okay. Thank you. Okay. So, what are you seeing here is actually the reason why you are seeing these differences. The image up is actually FTIR comparison of two extracts. And what you're seeing are different bonds, vibration and bending of different bonds that correspond to the presence of different chemical compounds. And what you're seeing down, because FTIR, I don't know if you're familiar or not, is essentially qualitative method. So we wanted to see quantitatively if we are seeing the same differences. This is why we did GCMS. And what you're seeing here in these circles are essentially the differences of different classes of chemical compounds. So you can see, for example, that subspecies sativa, A is always sativa, B is always indica. You're seeing that subspecies sativa, for example, contains more alcohols and more sulfur-containing compounds or mercaptanes. And you can see that you're having more carbonyl compounds in the subspecies indica. These differences are again the reason why you are getting different nanoparticles from different subspecies. Okay? So, few more things are going to be different that are also important for the interaction between nanoparticles and plants. What you're seeing here is the XRD image of our iron nanoparticles, and you can see that the iron nanoparticles synthesized from subspecies sativa were more crystalline compared to the subspecies compared to the nanoparticles synthesized from subspecies indica. That will also influence their local solubility, which will also influence their bioavailability to the plants. So everything is kind of connected. What you're seeing down is, are the results for the manganese nanoparticles. So what you're seeing here is again manganese from nitrate, synthesized from nitrate from subspecies sativa, manganese synthesized from nitrate from subspecies indica, and the same thing for subspecies sativa. What you're seeing is that you are actually getting far less soluble nanoparticles from subspecies indica compared to subspecies sativa. So, why is this all important? Because when people are synthesizing nanoparticles from plants, they tend to remove the plant residue. Now, we were wondering, is that smart or not? Do we need to remove plant residue or not? What you're seeing here is again FTIR, but this time of the nanoparticles that we were comparing, so iron nanoparticles from subspecies indica and iron nanoparticles from subspecies sativa, compared with commercial iron nanoparticles. And you're seeing that you have a whole forest of different chemical compounds that can aid to your bioactivity, that can help bioactivity of nanoparticles in the plants. You're seeing that you have a bunch of carbonyl compounds, you have a bunch of ethers and esters, you have amines, you have carboxylic acid, and so on. So, another reason why you shouldn't remove them, we were wondering if we don't remove them, will we see the differences between our biosynthesized nanoparticles and the nanoparticles that we purchase, so commercially available nanoparticles. What you're seeing here is exactly that. So first graph again ssativa, second graph again indica nanoparticles. You're seeing that ours nanoparticles synthesized from the plants without removing plant residue showed better activity compared to the commercial ones, but also better activity compared to the ionic form, because you also don't want to be making nanoform if there is no need for it. So if you can provide everything by using salt, why would you synthesize nano? This is why, because nanoform is giving you better results than both ionic form, and in this case, our biosynthesized nanoparticles were better in elevating the biomass of the plant compared to the commercial ones. Now, again, we analyzed the whole bunch of biochemical data, I think around 14 of them, but for the sake of time, I'm just showing you a few of them. So chlorophyll content is also increased more with biosynthesized nanoparticles compared to both commercially available ones and to the ionic form. This is iron acetate here because this is the salt that we were using for the synthesis of the nanoparticles. However, you can also see that the extract themselves, because we were wondering what part of our activity comes from the plant extract, so we also tested the extract themselves. You can see that they are also showing some activity, and you can see that we tested everything each week. Why did we do that? Because a lot of parameters tend to sort of flatten. Plants tend to reach homeostasis if you are testing them after, let's say, 3 weeks or a month. So this is why we tested them every week to see after which time plants will show the highest increase in activity. So we are seeing both increased chlorophyll content and increased antioxidant content with our biosynthesized iron, which again translates to more nutritive value of your soybean, because this whole experiment was done on the soybean. Now, this is another positive or not so positive thing, depending what you want to achieve. is that you would have to spray plants less. So all of this data is for foliar application. So we essentially sprayed nanoparticles on the leaves. Now we sprayed them only once at the beginning of our experiment. And what you're seeing here is translocation factor from shoots to roots. And you can see that our green nanoparticles are actually transported slower than the commercial nanoparticles, and no surprise, slower than the ionic ones that are transported pretty fast. Why is that important? Okay, so you're seeing the same thing for manganese. However, what I wanted to point out that these are the data that compare again the different salt of the manganese and their effect on the plant, and you will see that the antioxidant content and the total polyphenolic content will going to be very different depending on the salt that you are using. So here you are seeing the total chlorophyll content, and here you are seeing the antioxidant content, and you can see that their response to the either manganese synthesized from nitrate or manganese synthesized from sulfate is different. And the reason why is because you will have different impurities. You will also have the influence of ions. So if somebody asks me how would they need to make their nanoparticles, what I would tell them is that they need to select their subspecies of their plant, depending on the characteristic that they want to achieve. Because if you want to achieve smaller, you would use indica and so on. Another thing, depending on what you want to do. If you want to, for example, use them as pesticides, you would want something that has higher translocation factor from shoots to roots. In this case, you would want to use the other subspecies. And here you also need to choose very wisely the salt that you want to use, because again, depending on the characteristic that you want to enhance, you would use different salt for synthesis. Of course, I haven't done all of this alone, so I want to thank my team from the CIAS. And thank you all for your attention. Nardi.
Time for questions.
Just a question, I guess, about the pH of the different subspecies. So are these within a range commonly found in other plant species? Or have you looked at plants that have very diverse pH ranges and you see a difference there too?
These pHs, sorry, I wasn't clear. These pHs are for the synthesis part of the nanoparticles, so you need to adjust the pH. pH of the plant extract will also be changed. pH of our soybean extract was 6.13. However, if you're trying to take them to make, for example, your nanoparticles from tomato or from some other plant, the original pH of the plant extract will be different, yes.
More questions?
A very beautiful presentation and a very nice comparison that we can see while using different cultivars. One question that I have is, did you try to characterize the nanoparticles? Because this is out of my experience that I'm telling you, that sometimes what we perceive with respect to the nanoparticles, we say that these are oxides.
Oh, yes, I'm sorry. I forgot to mention that. I was kind of getting into all of that. Yes, these nanoparticles that I was mentioning are actually, excellent question, are actually the combination of iron 2 plus and iron 3 plus and manganese 2 plus and manganese 3 plus. And we actually determined that from the same XRD images that I showed. Thank you for asking that question.
So all the plants can be used as extract for nanoparticle preparation. This is just a query.
That is the question that I get the most. Excellent question. I can tell you for soybean, for tomato, and for wheat. I cannot tell you for the other ones because I haven't tried. I'm guessing many of them can, depending on the presence of the compound that have reductive and capping capabilities, which would be a lot of, for example, terpenes, a lot of alkaloids, a lot of polyphenols. If they have those classes of the compound, then yes.
So it may not be the subspecies, it may be species also.
First of all, it's species, definitely it's species, but also subspecies will play the difference. So you kind of need to account for both of them.
Okay, thank you, Milica, and thank everybody.
So we'll move to students' presentation now, which is there for cash prize to win. Our next presenter is Amrita from University of Calicut, and she is going to speak on seed priming and some tolerance in rice. Amrita?
Sir, I'm here.
Okay. Over to you then. So the timings are 12 plus 3, right?
So, good afternoon, everyone. My name is Amrita, and I'm a PhD student from the Son University. And my work is co-guided by Professor Josti Putur, Professor Dangar, and Professor Kishore Sridharan from the Son University. So, my topic is seed priming with GC3 and 4 nanosheets doped with essential elements can enhance salinity tolerance in rice. So, moving to the introduction, like we all know that salinity has a potential to affect the agricultural land worldwide, and it destroys the cultivated areas, like 6% of the cultivated area, and it adds 1 to 2% every year, reducing the global production of maize, rice, wheat yields. And rice is a glycophyte and is susceptible to saline soil, and there are various strategies that have been used against the salinity, and priming is one of them. Like we have known about many of the bulk priming. Now I'm going to talk about the nano priming. So stress tolerance and performance under salinity can be improved by nanostructured material priming. So why did I selected graphitic carbon nitrate? So urea is the precursor which has shown its ability against salinity, and it can resist the attacks of heat, strong acids and alkalinity based on some of the research that I referred. And it is non-toxic, metal-free, visible light-driven, amiable for large-scale production because the precursor is urea, and it has UV protection ability, and it is one of the least studied nanomaterial for their potential to act as a priming agent. And doping with essential elements can increase the priming potential of the particular material. So moving to the preparation and characterization of the nanosheets. So we prepared it from urea based on the thermal method, and we got both the graphitic carbonate as well as the doped one. From here onwards, I will refer the doped one as DGC3 and 4 and the other one as just GC3N. Okay, then in the XRD pattern, we couldn't get much peak on the degree in the doped one, which may be reflect that a small amount has been doped on the material. And however, when we did the TEM, we could get the clear detection that the doped material only contains the doped essential element, not any other contamination in that. Then moving to the plant material, seed priming treatment and growth condition. So we used Oryza sativa indica Lavanya species for the investigation, and nano suspension were prepared by sonicating both the nascent GCN and the doped GCN in deionized water 3 hours just before the priming. And sterilized seeds were treated with the GCN and DGCN, like 250 microgram per liter was used as the stress priming imparting concentration, and 12 hour was used as the priming imparting time. And the seeds were allowed to germinate and grow from there on a half-X Hoagland solution with and without 100 millimolar NaCl. So the standardization experiments yielded the stress determining concentration as well as the priming concentrations. Okay, again. So you have seen some of my slides. Okay, fine. So moving to the determination of growth biomarkers. Okay, so first I would like to tell about these were the impact of the seed priming on the stress and non-stressed condition that were given. So basically I have 6 study materials, that is control, and GC3 and prime plants without NaCl, and DGC3 and prime plants without NaCl, and the plants that have given the NaCl stress, and NaCl plus GC3N and NaCl plus SED and GC3N, and you can clearly see the height difference and root length difference in the picture itself. Then moving to the determination of growth biomarkers, it was clearly evident that from the very first germination percentage itself, there were clear indication that the salinity decreased the germination percentage and priming increases the germination percentage, which then came to shown in fresh weight as well as dry weight. So the nascent and coated one, both under control and stress condition, had a positive effect on the salinity. Then moving to the estimation of photosynthetic pigment, I used chlorophyll and carotenoid detection, and the pigment contents were also enhanced by both the nascent and coated GCN, both under control and stressed condition, which the significant increase was shown in coated GCN. The increase in coated GCN can be correlated as zinc act as a cofactor of sodium dismutase, which will protect the plants, mainly the chloroplast, and the negative from the negative effect of the ROS. Like from the ICP result, which I am going to show, it will be clear that the zinc content was very high in the treatments that I was given. And moving to the chlorophyll fluorescent parameters, like I selected Fe bar Fm, like the maximum quantum yield of PS2 as well as the electron transport flux of PS2. Like in both of the parameters, in stress it was decreased and in priming it was increased. Again, it could be attributed to the presence of increased presence of ascorbate and this SOD, which I got in my research. Then these are just the depiction of what I shown you earlier. Like in stress, you can see that AT0 bar CSM is decreased very low, and it has been gradually increasing with the treatment. And then moving to the quantitative estimation of MGA and electrolyte leakage, you can clearly see that MGA has been increased more than 50%, and which has been decreased by the priming, and similar result can be seen in electrolyte leakage as well. And similar result can be also seen in H2O2 and superoxide. And we can see a clear indication of a decrease in this content when we applied the priming, both in control and saline condition. And estimation of secondary metabolites and non-enzymatic antioxidants. Proline, we received like a peak increase in proline in saline condition, and which has been decreased when I give the priming. And similar case in glutathione as well as ascorbate as well. Sorry, for the glutathione and ascorbate, we got a clear indication of increase in glutathione and ascorbate. Like they are non-enzymatic oxidants which help us to resist the stress attack. So those things were increased, and it is less in saline treated condition. And moving to anthocyanin and flavonoid, the similar result were got. Like these things help the plants to compact with the stress they are getting. So anthocyanin content and flavonoid contents were really high in the plants that were got the priming. And moving to sodium and sodium potassium ratio, the sodium content decreased in the there was a high amount of sodium in the sodium only given, like the NaCl stressed plants, which has been decreased when we given the priming. And similar result has been shown in sodium potassium ratio, which means potassium was high in the treated plants. And these are some of the quantitative elements that estimation of elements that I did with the ICP OES. And we can clearly see that DGCN and NaCl plus GCN and GCN were having the most number of essential elements in it. And we also done differential gene expression for some of the genes like NHX, SOS, HMA, TIP and PIP, and actin was used as a housekeeping gene. And in SOS, clearly we can see that SOS gene is high in saline treated one, and it has been decreased when we were given with the priming. And we also tried to study whether if we my study was like a culture rack study or like a kind of closed chamber study, right? So I tried to go out from the chamber to the port, and we could clearly see some fertilization effect on that. And we plan to next step, we plan to do the transcriptome and metabolome analysis. Like we already done with the transcriptome analysis, the data to be analyzed, and metabolome analysis we have given. So when we get all those results, we will get a holistic idea. So what is happening and what this priming is giving, so that we are getting this much good result. And future plans continue the field and port study with and without NaCl. So conclusion, priming with niacin and coated GCN had a fertilization effect and increased salinity tolerance in rice. Priming with niacin and coated GCN reduced negative effect of salinity on photosynthetic pigment and enhanced membrane stability. GSH, ascorbate, anthocyanin, flavonoid, whatever you name it, I have done already almost all the biochemical analysis, and everything has shown us some good result itself. And overall GCN coated with micro element is a good strategy for sustainable agriculture to increase abiotic stress tolerance in crop. And let me acknowledge my lab members like Putur lab members and Prakash lab members. I also want to thank Connecticut agriculture team to help to done with the ICP OES, and funding was done by the CSIR Foundation and the Fulbright Foundation of USA. This much. Okay, thank you. This is the presentation. Okay.
Yeah, so we can take questions. Please.
Could you just explain a little more of the enzymes that you measured? HM, SOS, what are these? Just give us a little bit of background to them. Yes. Thank you.
Okay. So from here it starts like MDA. MDA is a indicator of stress, like malondialdehyde, which indicates whenever there is a degradation of the membrane, this thing will come. So if the stress is there, there will be more MDA. So that can be quantified with the help of malondialdehyde. And electrolyte leakage, whenever there is an membrane breakage, electrolytes will come out of the membrane, right? So we can quantify the amount of electrolyte leakage, and from that we will get whether the plant is stressed or not. Then moving to hydrogen peroxide and superoxide, I don't think we should explain it. Hydrogen peroxide and superoxide itself is an indicator of stress. Then proline is a thing that is, okay. Then glutathione ascorbate, okay, you are pro in that, I'm not going to explain it to you. Okay. Then anthocyanin and flavonoid. So these are also, okay. Okay. Okay, so like from the quantitative estimation of the elements, we were got with the zinc was increased in our treatments. So we selected the HMA too, so that we could see whether is this we didn't do the transcriptome analysis first, so we just selected some of the genes which were high based on our analysis on the biochemical analysis, and we selected that and we tried to see whether it is increased or not. So once we will done with the transcriptome analysis, we will get a holistic idea and we will select some of the genes more particularly. Here what we did that from the biochemical analysis, what we received, we selected some of the genes, and SOS is a salt overlay sensitive one, which means whenever there is a salinity, that will be high and it will be more activated, and and when priming is given, it is decreased. And HMA similarly, whenever there is a zinc transport is there, we can see more like HMA. And NHX is a sodium high affinity potassium channel itself. So I suppose that there is more electrolyte leakage in stress, right? So that may be the reason why NHX is high in the stressed one. And TIP and PIP are some aquaporins and tonoplast protein, right? So I suppose it that way, because of the stress, these things are increased, and when the plants are encountering less stress because of the priming, those things are reduced. That's what I suppose. Yeah.
Any other question?
Yes.
Question here.
Yeah.
So wonderful talk. I'm convinced priming is very beneficial. My question to you, what happened to the chemistry of your graphitic carbonatrate? What happened? Are they still on the seed, or they degrade into different form of chemical into your seed? What happened to graphitic carbohydrate, nitrate?
That thing we are yet to study. Like I plan to tag it with some fluorescent dye, and whether it is going into the plant or not, I have to study it. But what I am the thing what is going inside my head, or what I discussed with my professors, is that so we are giving the seed priming, right? So this material might be coating the seed and protecting it from the negative effect of salinity, which will help them to grow better in the early stages of their life cycle, that is imparting to the next stages of their life cycle. And sometimes the coated one which is going into the whole gland media that we are giving, this thing has many pores in that, so NaCl can be absorbed into those small pores which might be giving less sodium to the plants. That what we speculate. There is no much reference so that we can go and check in the publication area. So I think we are some of the pioneers who is working on the salinity and GC3N. So more analysis is needed, like transcriptome, metabolome, and this fluorescent tagging. Everything comes together, then we will get a holistic idea. So what is happening inside the seed, or whether it is going into the plant or not.
Okay. Any other question? Because then I will ask one.
Okay.
So, in your wild type control, untreated, unstressed plant versus the treated one, stressed and unstressed. When you check ROS, H2, O2, MDA, EL, they are almost similar level. But if you look at the state of plant seedlings, control plants are way healthier than the treated plant with these chemicals. Why so? Are you getting my question? Can you take me to the...
Yeah. It will. The system is little slow.
It's okay. It's okay.
It will. Okay.
Yeah. So,
Apart from that, everything came.
here.
Okay, fine.
So if you look at the control plants versus NaCl, DG, C3 and 4 plants, they don't look same. Obviously,
the first and
the last
second
one.
in the,
Yeah.
this one?
No, the last one on the bottom panel. Okay. If you come to the MDA content, let's say, or the other ROS, they are similar or even less. But the plant, stress plant still looks stressed. They should have more MDA content. They should have more electrolyte leakage. Next slide. Ideally, they should have more peroxide, superoxide. But in your case, it is almost similar or less. So what could be the reason?
Like from what the result I got, like what I speculate is that might that plan B is not getting that stressed. Like even though it is in stress, it is not experiencing that much stress, or it might be like I told you earlier, right? The GCN that might be disintegrated from the primed seeds, which is going into the whole gland solution, might be absorbing the NaCl and other things from the solution, like the sodium and chloride, or some of those things which is high in those, and they are absorbing it, which might be reducing the Na plus element which is presenting in the whole gland, which might be giving less stress than the other one. And I don't know whether the control plants were that stressed or not. Like we tried it couple of times, these similar kind of result were getting. Like I repeated the same experiment for at least 4 times, similar kind of result were getting.
Controls are non-stressed. Yeah. But so control first versus, let's say, last. So there not much change.
There is a slighter increase, but due to the stress is having high, it is not
your
that
time
much
is
short.
up. Yeah. Okay, you have answered. Right.
Okay, now we move to Iba Habib from the University of Calicut with the presentation Go Green, Valorization of Fruit Rind into Nanofactories.
Good afternoon, everybody. Our lifestyle is changing, our eating habits are changing, we are becoming more unhealthy. So to adopt a healthy lifestyle, we try to incorporate fruits and vegetables in our diet. We're only bothered about the inner juicy part or the inner tasty part of the fruit. We are never bothered about what happens. Is being wasted. So it is a hectic issue for our country. Similarly, as per the Ministry of Food Processing Industry, around 12 to 21 billion tons of fruit and vegetable losses are happening. This is also a problem. So it's really critical that we have to come forward with some method to utilize these waste into something valuable. This shows that around 42% of these vegetable and fruit peel waste are coming from houses, while 38%. We know that fruits are rich in antioxidants, phytochemicals. That's why we make it a part of. That's why we make it as a part of our diet. But like fruits are rich in these substances, its peel is also rich in them. It's also rich in bioactives like phenolic compounds, fibers, polysaccharides, and the major ones that are found in fruit peels are antioxidants, pectins, flavonoids, polyphenols, which can act as pharmaceutical, nutraceutical, or can be developed into a functional food, therapeutic, and so on. So from this slide, I can analyze or I can conclude that fruit rinds are a hidden treasure. It itself is a nanofactory that is enriched with tailored capabilities. But before converting this waste into something wonder, we have to know or have an idea about what is green nanotechnology, or nanotechnology for green innovation. It is a cleaning technology that aims to wipe out the environmental and health risk associated with the conventional production of nanomaterials by enhancing the means of producing nanomaterials in an eco-friendly means that sustains the that enhance the sustainability of the environment. That means mainly by this one we aim to produce nanomaterials with renewable recipes instead of toxic ingredients in a cost-effective and energy-efficient manner. Under green nanotechnology, what we aim is to synthesize nanoparticles using plant materials in a biological means, because it is considered to be more beneficial, more eco-friendly, cost-effective. It has high reproducibility, more intrinsic stability, and it's our safest alternative to the sustainable nanosynthesis. Nanoparticle synthesis. Now we're moving to nanoparticle synthesis. Mainly, as per reports and as per literature, there are two types of nanoparticle synthesis. Two approaches are followed. One is bottom-up approach, and other one is the top-down approach. In top-down approach, we deals with physical and chemical means of synthesis, where physical requires huge amount of atmospheric pressure, and it takes long time to achieve a thermal stability. It have its own limitations. And here, nanoparticles are synthesized from bulk material. Now, in chemical method means we use toxic chemicals. And what happened to these toxic chemicals is like, whenever we are using a toxic chemical, there will be byproducts. But these byproducts are hazardous such that we can't decompose it and it poses threat to the environment. Here methods like chemical reduction and co-precipitation are followed to synthesize nanoparticles. But under bottom-up approach, where single atoms or molecules are used for synthesizing nanoparticles, here biological approach or biological method is followed, where we use plants and its parts or microorganisms like fungi, bacteria, algae. Here I'm dealing with plants. Each part of the plant is used, or as a plant whole is used. But my approach or my initiative is to synthesize nanoparticles, that too metal nanoparticles from fruit rind. We know that nanoparticles are mainly of two types, inorganic and organic. Organic ones are carbon-based, while inorganic ones are metal or metal oxide based. And these metal nanoparticles, among them, silver nanoparticles are the ones that are considered to be more superior because of its low toxicity profile, its higher electric conductivity, conductance, and it has chemical stability at ambient settings and all. Now, my study is about the green synthesis of silver nanoparticles. The fruit peels, what happened is that initially we collect the fruit peels, we dry it, and an extract, aqueous extract will be prepared, which is mixed with silver nitrate solution in a specific ratio at optimum conditions of pH, temperature, and the color change will be indicating the nanoparticle synthesis. That means when we combine metal salt solution with plant extract, biogenic nanoparticles can be synthesized. These nanoparticle synthesis mainly can be divided, its mechanism can be divided into 3 phases. As phase 1, where nucleation and reduction occurs, where the silver ions are reduced by the phytochemicals present in the plant extract. I earlier about chemical synthesis, where we use chemical agents. We have to use reducing agent, caping agent, and stabilizing agent. But when we are using a biological extract, that means a plant extract, the major advantage is that we don't have to use these kind of reducing, capping, or stabilizing agents separately. Instead, the phytochemicals that are present in the plant extract itself act as these, and they bring about the reduction and nucleation. In the second phase, once these silver ions become silver atoms, they aggregate and increase in stability and size to form larger ones. And it occurs based on the effect of pH and temperature. Finally, when it has attained a stable size, it will be stabilized by the caping agents that's present in the plant extract, and you will get the nanoparticles. Now let's move on to a real world example. This was done by me, and here nanoparticles is produced from jackfruit peel. The variety of jackfruit that was chosen for this is the wild jackfruit, which is known by the scientific name Atrocarpus hirsutus. The ripened fruits were collected, the peel was collected, it was shade dried, aqueous extract was made, it was mixed with silver nitrate in a ratio of 1:9. A dark brown color was observed. This indicated that nanoparticles were formed, which was confirmed with the help of UV vis analysis, where when we get a surface plasmon resonance peak in a range of 400 to 500 nanometer, we can confirm that silver nanoparticles are being formed. And this happens, or this brown color occurs, due to the ability of these chemicals, phytochemicals present in this extract, to reduce the monovalent silver in the silver nitrate to zero valent silver nanoparticles. Once we have confirmed that nanoparticles are formed, we centrifuge it. Physiochemical characterizations are carried out. The characterizations carried out were SEM analysis, which is often done to study the size and shape of nanoparticles. Here the nanoparticles were spherical in shape with a particle size range of 19.53 nanometer to 26.04 nanometer. And XRD analysis was done to confirm the composition, to understand the chemical composition of these nanoparticles and the crystalline nature. The nanoparticles were nanocrystalline. It was confirmed to be silver based on the 4 Bragg's reflection peaks as per the ICD data. And here the crystalline size, average crystalline size, was found to be 22.14 nanometer, which as per the Scherer equation. Finally, dynamic light scattering or DLS analysis was carried out to show its stability over time, its agglomeration state, or the what kind its PDI, and this PDI indicated that these nanoparticles are monodisperse without any aggregation. Not only that, with these nanoparticles, I have carried out catalytic degradation of organic dyes. We know that our water bodies are presently polluted with the dyes or the chemical pollutants that are released from textiles and printing industries. There are conventional methods to treat them and reduce them, but these methods have certain limitations. The major limitations they have is that they produce hazardous components and they are highly expensive. These hazardous substances cannot be degraded. But by using this method, where we are using a nanocatalyst, what happens is that it can be completely degraded and it's safe. This catalytic degradation of organic dyes with the help of silver nanoparticles are a reductive degradation using nanocatalyst, and it can be considered or it can be proposed as a safest alternative for wastewater purification. Here in this experiment, I used a reducing agent, that is a sodium borohydrate. 2 organic dyes were chosen. One is methylene blue, other one is methyl orange. And a stock solution of biogenic silver nanoparticles of 1 milligram per ml was also prepared. Now let's move on to the experiment. 2 reactions were carried out. One is an uncatalyzed reaction, other one is a catalyzed reaction. Under uncatalyzed reaction is like we'll be taking just the dye and the sodium borohydrate. You can find that normally this methylene blue, which is a toxic carcinogenic dye, have an absorption peak at 664 nanometer. It's clearly visible, 664 nanometer. But, what happens, it's nanometer, 664 nanometer, and in this case, there's not much reduction or degradation of the dye has not taken place. But when you look into catalyzed reaction, means we have added silver nanoparticles as a catalyst. What happened is that a huge change has taken place after, it's a time dependent reaction. For initially in the 1 minute, only around 8% was degraded. But when it became 5%, around 32% was degraded. And around 10 minutes, whole the substance was degraded. Second is methyl orange. Here also what happened is that same study was carried out, and this reaction follows a pseudo-phosphotokinetics. And sorry for just few minutes, I'll complete now. In here also you could find that it took 5 minutes completely for doing this degradation. Hence, I conclude that we should remember the fact, we should understand that fruit rinds are more than a waste. They are a sustainable source for nanoparticle synthesis, and this act as themselves nanofactories as a solution for the 2 major problems, that is waste, pollution, and resource scarcity, because when we are using fruit peels, we don't have to depend upon the plants, and it will be beneficial. And if we follow these strategies, we can incorporate green nanotechnology, which is our real future. Finally, I conclude by asking you all to reimagine, or requesting you all to reimagine waste, this fruit peels, as the key for a sustainable future by adopting the notion of reduce, reuse, and recycle into our lives so that we can create a happier earth with a better life. Thank you.
Time for questions.
Yeah.
Please, Baoshan.
Yes. So for your synthesized silver nanoparticle, when you try to degrade methylene blue, so what happened to your particle? Are they recyclable, or they become a silver ion? Then that's a heavy metal if you release into the environment. What happened to your silver nanoparticles?
When the silver nanoparticles are used for this catalytic degradation, they can be recycled. I have tested that too, because they are not just released into the environment. We can use it again and again.
So you have to use your biological materials, then resynthesize, reduce.
Not like that, sir. Initially, the plant extract and metal salt will be reacted to form nanoparticles. We'll be adding it as nanocatalyst for the reaction medium. After, once the degradation has carried out, we'll be centrifuging that solution such that we can collect these nanoparticles, because they often sediment out. When we centrifuge, we'll separate it, and this same nanoparticle can be used again for carrying out this reaction.
More questions? Liz?
I suggest you work out what the number of times you can reuse it is. That would be quite a good experiment. You said that the diameter of your silver nanoparticles was between 3 and 24 nanometers, but they can't spontaneously appear at 3 nanometers. So would you like to suggest an alternative? What's actually happening there?
I didn't get to.
So you say that the diameter of your nanoparticles is between 3 and 24 nanometers?
No, ma'am, 19.53.
Okay, 19.53, but it's the same concept. They can't just spontaneously appear at 1.93. What's actually happening?
That's like the metal salt solution, that is silver nitrate, plant extract will be reacted in a ratio of 1:9. And once we observe the color change, UV spec will be done, we'll confirm it. And
okay.
So, ma'am, for the completion of this whole reaction, we are incubating it.
Okay, but I guess what's the resolution of your transmission electron microscope? What's the minimum particle size you can visualize?
Like 10. It's even very smaller particles can be
very small. Less than 1.9 nanometers?
I don't know.
Okay, I just suggest that it's the resolution of your microscope that's limiting your lower limit, not that you're seeing spontaneously 1.9 nanometer diameter appearing. We can talk.
Okay.
Good presentation, Hiba. Wanted to know with regards to why did you choose only Hirsutus and not Artocarpus heterocarpus with respect to the production and the wastage generated?
No, it's like there are several reports in the other varieties of jackfruit. This is a rare variety and it's a wild one. That's why I choose this plant.
The waste that is generated by Hirsutus is more than...
No, not like that. The more waste will be, it is a very small sized fruit. So less waste will be generated. If you take the large one, Atrocarpus heterophyllus, the big jackfruit, more waste will be generated. Same procedure can be applied to them and same nanoparticles can be synthesized.
Okay, thank you.
Yeah, unless you generate that, it is just a speculation. In your dye degradation experiment, what did you take any other form of these silver nitrate, some other formulation of silver nitrate nanoparticles, which could be, let's say, used for comparative studies, like your nanoparticles were better or,
No, I have only used the nanoparticles that I have synthesized, biosynthesized ones. And this is just a preliminary study for it's a part of my PhD work. Only the nanoparticles synthesized by myself was observed, and I could find this activity I have to perform comparing with the standard purchased nanoparticles so that to look what its efficiency.
Okay, thank you. Applause. A quick announcement for the poster sessions. All the students that have a poster are welcome to stand in front of it and be questioned by the judges for the poster award. So please, during the poster session, be at your poster. So now we move to Erin Lincoln from the Merrimack College, and she will talk about understanding silver nanoparticles interaction with essential elements and heavy metals in Solanum lycopodium.
For the past year, and I'm currently in a one-year master's program. The work that I'll be presenting today focuses on the work I completed as an undergraduate over the last year. So nanomaterials in general are substances with a size between 1 and 100 nanometers. There are a variety of metal nanomaterials in particular. Silver nanoparticles have really unique properties, including antibacterial and antifungal properties, as well as really high electric conductivity that makes them applicable for a variety of industries and products. These include food wrappings, textiles, particularly waterproof textiles, bandages and wound dressings, toothbrushes, and detergents, laundry detergents. Given that they have such a wide range of uses, approximately 500 tons of silver nanoparticles are produced globally each year. So silver nanoparticles can be released into the environment at any point during their synthesis, their consumer use, or their disposal. And this exposure in the environment can have negative effects at various levels. In cells, they can result in decreased membrane integrity as well as cell viability. In organisms such as mice, they show increased cortisol as well as central vein damage. And in ecosystems, particularly aquatic ecosystems, we see the inhibition of bacterial denitrification. Silver nanoparticles can be used to promote plant health in combination with other elements and certain treatments. However, at high concentrations, they can have toxic effects. When pesticides or fertilizers containing silver nanoparticles are used on crops, there's the potential for them to be accumulated in plants and even translocated into fruits, either in the particulate form or in the ionic form after they are oxidized. So we have three key questions that we're focusing on in this research. Mainly, what plant organs are silver nanoparticles accumulated in, and are they translocated into fruits? Secondly, do silver nanoparticles interact with essential elements and heavy metals within plants? And thirdly, do silver nanoparticles interact with vascular tissues as they are translocated through plants? For our materials and methods, plants were exposed to 30 milligrams per liter of either silver nanoparticles or silver nitrate. After 30 or 70 days of exposure, our plants were harvested per organ, and we split each organ into upper and lower sections so we could compare the younger and older parts of the plant. And our samples were prepared for elemental analysis using ICP-MS. I'm going to split this talk into three sections, the first focusing on the translocation of silver, the second focusing on the interactions between silver nanoparticles and essential elements as well as heavy metals, and the final part focusing on the impact of silver nanoparticles on xylem cells. So this is a diagram I've created showing the overall accumulation of silver after they've been exposed for 70 days. The red color is the highest concentrations of silver we see, while the green color is lower concentrations. And now I'm going to break it down by each plant organ, starting with leaves. For both our silver nanoparticle and our silver nitrate groups, we see that silver is very highly accumulated in the upper, younger leaves, a lot more so than in the older leaves on the bottom. And that is significant differences there. We see a similar trend for our nanoparticle group in roots as well, and we see significantly more silver accumulated in the lower roots of our our nanoparticle group compared to our nitrate group. Additionally, we are seeing accumulation of silver in fruits for both our silver nanoparticle and our silver nitrate groups, which is very concerning. Overall, we're seeing silver nanoparticles act as a mobile element and be heavily translocated into the younger parts of the plants, including leaves, roots, and fruits. After this harvest was completed, we started to wonder how these silver nanoparticles were being translocated through the plant and all the way into fruits. So we looked at different membrane transporters. Previous research done by both our lab and other labs have indicated the role of aquaporin channels in the uptake of silver nanoparticles. Aquaporins are mainly used for the uptake of water. So our next harvest compared two genotypes, the first being our wild type plants and the second being a mutant plant that was knocked out for our aquaporin channels. Again, this is kind of an overall result of our silver accumulation after 30 days of exposure for both wild type and aquaporin knockout plants. In leaves, we see significantly more silver accumulated in our nitrate group for our wild type genotype compared to our aqua nitrate group. However, we're not seeing any significant differences between our wild type and aqua genotypes in the nanoparticle group. Where we do see significant differences in our aqua nanoparticle group is in the accumulation of silver in roots. However, we're actually seeing silver nanoparticles accumulate further in the roots of our aqua plants as opposed to our wild type plants, which is kind of the opposite of what we expected. So overall, we see that our wild type plants exposed to nanoparticles accumulate more silver in their shoots, while our aqua plants accumulate more silver in their roots. However, overall, we are seeing our aqua plants actually accumulate more silver than our wild type genotype. So we can infer that there are other membrane transporters involved in the translocation and uptake of silver nanoparticles. Next, I'll be talking about the interactions between silver nanoparticles and other elements. Again, previous research has indicated that silver nanoparticles can interact with both essential elements and heavy metals. So we chose 6 different essential elements and 6 different heavy metals to focus on. So after 30 days of exposure, these are the correlations we got, mainly between silver and these other elements. We see a lot more positive correlations in our wild-type genotype for both our nanoparticle and nitrate groups, which indicates that silver nanoparticles, or silver in general, is interacting a lot more with other elements in our wild-type genotype. Where we see similarities between our wild-type and aquaporin groups for our nanoparticle-exposed group is positive correlations between iron and silver, and positive correlations between copper and silver. So as the concentration of silver increases, the concentrations of iron and copper also increase. We also see a lot of positive correlations with heavy metals, mainly in both of our particulate groups for wild type and aqua. We see strong positive correlations between silver and vanadium and silver and cobalt. So as our concentrations of silver are increasing, the concentrations of vanadium and cobalt are increasing as well, which is very concerning. Lastly, we'll be looking at the impact of silver nanoparticles on xylem cells. This is a continuation of previous work that was done in our lab. For the harvesting and sectioning of our xylem cells, we used 2 centimeters above the soil line. Now, after our 70-day exposure, which again was just our wild-type genotype, we saw a significant decrease in xylem cell size in our nanoparticle group compared to both our control and our nitrate groups. However, we did not see this in the 30-day harvest for either our wild-type or aqua plants. What we did see is that all of our aqua groups had significantly smaller xylem cell area compared to our wild-type groups. This is likely because the uptake of water is reduced in our aqua knockout because our aquaporin channels are knocked out. So overall, we see silver nanoparticles act as a mobile element while they are translocated through plants. We see silver nanoparticles translocated into fruits. Essential elements, particularly iron and copper, as well as heavy metals, vanadium and cobalt, have positive correlations with the concentration of silver nanoparticles in plants. And finally, silver nanoparticles cause significant decreases in the size of xylem cells in plant stems. For future work, we'll be focusing on molecular analysis and looking at the expression of other membrane transporters outside of aquaporins. We'll also be focusing on the detection of nanomaterials and particulate silver levels in plants, and finally looking at root architecture and how vertical and lateral root growth are impacted by silver nanoparticles. I'd like to thank all the members of my lab, particularly Dr. Nori, as well as the lab managers at Merrimack College. I'd also like to note that all illustrations were my original work in this presentation. And if you are interested in collaborating on any kind of scientific illustration or graphical abstract, feel free to reach out to my email. Thank you.
Questions?
Sorry, always. Fantastic. Thank you very much, Erin. Do you think it's possible to distinguish between the silver concentration inside the root and what's stuck on the outside of the root? Do you think that's possible?
So we did wash our roots pretty thoroughly, but that's not necessarily a guarantee that we're getting what's stuck off of the outside. So we would have to look into potential ways that we could distinguish that.
Okay, thanks.
Great job, Erin. I have about 10 questions, but I'll ask most of them later.
Right.
So I think the microbiome in the rhizosphere and within plants is obviously very important. What do you think the impact of the silver nanoparticles versus the silver nitrate is on, say, the rhizosphere microbiome?
I'm not entirely sure. We did also analyze soil samples, and we saw pretty similar levels in both our silver nanoparticles and nitrate. There weren't any huge differences that stood out, but there's definitely further work that we could do on that.
Yeah. So you checked the aquaporin knockouts. So whether those are, which family of aquaporins you have selected for making mutants?
PIPs, so the plasma membrane transporters.
So you have made all PIP mutants?
Yes, all of them were knocked out.
So all were giving the same type of results?
Could you repeat that?
So I'm asking that whether you have used all knockouts of PIPs?
Yes, yeah. They were all knocked out in one mutant.
So it's like 10 genes are there, so you knocked out all of them?
Yes.
Okay. So, okay. So these nanoparticles, since they are very small, so isn't that they can pass through the plasma membrane directly? Whether they require aquaporins?
They require some kind of membrane transporter.
Okay.
So that's kind of our next step, is to figure out what exactly those membrane transporters are.
Yeah, thank you.
Can I extend on that one? I think maybe because if you knock down all aquaporins, 10 aquaporins, I don't think the plant, a tomato will be able to grow. I think you might have few aquaporins, 1 or 2 knocked down. It's almost impossible that all 10 aquaporins will be knocked down and in a single line.
That makes sense. I would have to talk with Dr. Nori about the specifics of that. Again, we did acquire these. We did not synthesize them ourselves.
Yeah.
Okay, we've concluded. An applause. So we finished this session. It was a pleasure chairing it. Thank you to all the speakers and to the audience.
The same here. It was pleasure chairing the sessions, and yeah, thank you all, to all the speakers.
And please now have a round of applause for our co-chairs for excellent job. Poster session is already on now at 4:20 to 6:20. I think we need to move on, and students specifically, please stay with your posters and to answer the questions, and there are prizes, and you are maybe lucky that competition is less. There are a few no-shows and 23 posters, and there will be three awards, so you have better chances. So please go and present your posters, and all the participants, please go and have this glance over the posters and enjoy those. Thank you.
പറയണമെന്ന് പറഞ്ഞു